So what kind of chronological periods are people focusing on for isotopic analysis? It's mostly Neolithic and Chalcolithic, as we can see here, and from the pie chart here. And this isn't really surprising because obviously Turkey is one of the main areas for domestication and it forms part of the northern part of the Fertile Crescent, domestication, the introduction of agriculture, sedentary populations. So obviously a lot of interest is in looking at diets and how animal management and dietary habits changed from that transition from sort of hunter-gatherers into sedentary agriculturalists. So it's not really surprising that we see this kind of pattern of a lot of research focus has been put into these kind of early Neolithic and also Chalcolithic periods with the development of farming. And again, similarly related to this, the most research has been done on human dietary habits and also animal management practices. So again, it's a similar kind of thing, looking at how maybe dietary habits or how people change their subsistence strategies, their agricultural strategies, or how things have changed over time. So it's sort of linked in with what I was talking before about the chronological period, this sort of what kind of research people are doing is related to this. We see that some themes, some sort of types of research are underrepresented. So things like weaning, arable agriculture, so actually direct isotopic analysis on plants, or that being the focus of the study, and also paleoenvironment reconstruction as well. And then this is sort of how the research is conducted. So there's three categories: holistic studies, and basically that was we counted the studies that use more than one type of analysis. We combined carbon and nitrogen, stable isotopes of carbon nitrogen, delta carbon 13, delta nitrogen 15 to be one. So if there was a paper included carbon nitrogen, they're usually done together. For dietary habits, that was just one. But if it involved something else like strontium or oxygen or trace elements or had something in depth about zooarchaeological analysis or physical anthropology or something, that was a holistic. We counted that as a holistic study. See, most studies have been conducted on single sites. There's very few studies that have looked at sort of wider patterns comparing different sites or different regions. Most isotopic studies tend to focus on that one particular site and people from that one site, or animals, or whatever it is they're studying. So if we move on to trace elements now. The trace elements actually started earlier in the 1980s, and they sort of had sort of mixed fortunes. It's kind of had a bit of a hiatus, and then in the early 2000s, it kind of begun again, and then sort of like peaked, and in the sort of like the last sort of decade as well, sort of similar to isotopes. But as you can see from this chart, it's not quite a sort of a linear increase. It's sort of been up and down a bit. And again, we looked at time periods, and sort of it's the opposite for trace elements. It's mostly been from sort of Hellenistic, Roman, Byzantine, post-Iron Age populations, and this is to do with because obviously trace elements are conducted on appetite from the bone, and this is more susceptible to diagenesis. So the more recent bones will have less susceptible to diagenesis, but in better condition. So this is often why trace elements have been applied to these populations from these time periods rather than earlier time periods. And when we look at what the themes of the research, it's similar to the isotopic analysis, mostly dietary habits, so looking at what people are eating, but also as trace elements, because the human body and the bones accumulate elements from their environment, things like heavy metal accumulation. So looking at how maybe the effects of sort of industry or something like manufacturing on a site will have affected humans, and sort of how the humans were affected by their environment and how that got into the systems. So this is sort of the two main themes. But again, it's mainly like the isotopes. It's mainly dietary habits. And when we again look at the sort of how things are done, again, it's very similar to the isotopes. Actually, it's a little bit holistic, but very few multi-site analysis, and it's mostly single sites that have been studied. There's also with trace elements, there's also an extra category, which is case studies. So there's been some sort of case studies into the methodology that have been done in Turkey. So to sort of have some conclusions, sort of talk a bit about some ideas about how we can move forwards as well, and how things are going. So first thing is, let's not forget about trace elements. Like we saw from the initial chart for the trace elements, that publication studies, which again means the number of research studies conducted, have kind of been up and down. Their fortunes have kind of come and waned and stuff. But it is a useful standalone method, and it's also very useful if you do it in conjunction with stable isotopes, especially for like diet, because you obviously have the two different parts of the bone that are looked at, especially for things like carbon nitrogen. So you have collagen for stable isotopes, and then the appetite for trace element analysis, and they have different dietary routing mechanisms, so we get a better overall picture of the diet. They can also complement each other, and they can also the collagen for stable isotopes, can be a sort of diagenesis control to see if the results we're getting for the trace elements are accurate or not. So long-term sustainability, how do we sort of keep going? And it comes down to three things really: what, where, and how. So first of all, the what, there needs to be a better understanding and awareness of the utility of biogeochemical methods, so isotopes and trace elements. So what are isotopes? What are trace elements? How can we use them? Where can we use them? What can we apply them to? And this can be done at all levels, from all the way from sort of undergrad student all the way up to sort of research institutes, universities, site directors, bureaucratic and funding levels. Just a sort of better awareness, I think, would be useful to help keep things going. Sort of where is the development of isotope labs, especially. There's a few places where we can do trace elements, but there's not so many places where isotopes can be reliably prepared and applied to bioarcheological organic materials, especially in Turkey at the moment, and development of further labs will allow for further research and a greater variety of research as well. And there needs to be a better, greater collaboration and cooperation between specialists and directors of projects, for example. Like we said before, a lot of projects are focused on singular sites or singular aspects of things. So it'd be better to sort of try and get these bigger pictures maybe going. It's more holistic approach. So how can we do this? So it sort of needs to be a better understanding and comprehension of how we can integrate isotopic and trace element studies into research, archaeological research in Turkey. This goes all the way from the beginning. So they should be considered at the very beginning of a project planning phase, for example. So we're going to do this, and we think we're going to find these, so therefore we want to do these kind of analysis in the future. Again, like I said, better cooperation between specialists, and also the access to data and the publishing of data. So a more robust and standardized way of publishing data, and also access to things like legacy data for later comparisons and stuff. So you want to compare data sets. It should be easier to do that. So there's a few steps that are being taken at the moment, and sort of just talk briefly about like how we can do this sort of actual practical terms. So like I said, mentioned before, training and teaching will be very important, at all levels. So this will enable this sort of long-term sustainability, which we want to see in Turkey. We want to see this kind of research increasing and continuing, and not kind of stuttering and stalling. And education. So like, again, the students, I think, are really important, especially for undergrad level, to have this training and teaching in biogeochemical analysis. Both in English, I think, is important, because unfortunately, English is the international, I mean, it's fortunate for me, but less fortunate for everyone else, is the international language of science. So it's very important that students understand these topics in English, but also, especially in Turkey, we should be also teaching in Turkish, or there should be resources available in Turkish to Turkish students, because after all, these Turkish students will be the next generation of researchers in Turkey working on Turkish material. And if they're not trained from an early age, then this is a potential stumbling block later down the line. So myself and Kamran Hoca have also been, to sort of address this, we've been publishing a few papers in Turkish, sort of like introduction to isotopes and how they can be used, what they can be used for, what they're applied to, how to interpret them, how to sample protocols, these kind of things, the methodology and the theories behind them. And there's also a project that I'm involved with, BioArcheology, which is it's a database and a website which will, when it's finished, hopefully in the next year or so, will contain an integrated database of all isotopic data from the Near East, so all the way from sort of Greece and the Balkans, all the way through to West Iran, South Caucasus, Egypt, Levant, Mesopotamia. And this will help, hopefully, to start addressing things like the issue of access to legacy data. And also, there will be sections of the website on things like how to sample, different kind of sampling or extraction protocols, preparation protocols for preparing your samples for analysis, and also sort of best practices, or how to report your data, how to create an isotope chart, or how to interpret these things and stuff. So that's it from me, and I'd like to say again, thank you to the organizers, thank you very much, and thank you to all of you for listening, and I'm happy to take any questions or any discussions now or across the course of the day. Thank you very much.
Thank you very much. This was an extremely interesting review, let's say, of what's going on in Turkey and in general. Thank you, Dr. Kameray Özdemir, and thank you, Dr. Irvin. Are there any questions? Because I would not let the questions at the end. We lose the spirit. So if there are any questions at the moment, please go ahead.
It's not really a question-provoking talk, I guess, because there's no data to discuss. But I mean, if anyone at all across the course of the day wants to discuss things.
I do have two.
Okay, all right.
One is a comment and the second is a question.
Yeah.
The comment is that it is extremely interesting in your first slides to see that the papers increased the last years. I believe that one reason for this is that awareness is now present. But I believe that the second is that technology has been developed extremely rapidly. And now we have the ease of using this technology to be able to get data and publish data.
Yeah.
Therefore, we're on the lucky side because those years we do have the technology with us.
No, absolutely. The technology's improved massively, especially over the last decade. You now need like smaller samples compared to previously, which obviously helps because of Turkey, where it is geographically, it can be quite arid sometimes, so preservation isn't always great. So the small amounts of samples we can use, even better. Yeah. It's also become a lot cheaper, so it's become a lot more affordable for more projects. Because of smaller samples and better technology. So yeah, there are some other things, but I think you're right. It is a general, there has been an awareness that's been growing over time, especially last decade. People are like, oh, we can do this, and we can do that, and we want to do this, and we want to do that. But I really like to see that continue, because like I say, great work has been done so far. And like I say, we'll see some great talks today about new data. And it'd be a real shame if this kind of didn't continue.
And the question is very simple and short. Do you know which is the technology basically now that is being used for trace element detection?
Well, I'd have to hand that over to Kameray Hoca because he's the trace element expert. I do more isotopes.
Okay, thank you. Yeah, so we're much further in the area. Perfect. Thank you very much.
Right. Thank you very much again. Thank you, everyone.
And now I would like to call Dr. Donald Kale. I hope I pronounce it correctly. He's going to present a work that has been done by Semih Özen, Doruk Cafer Özgü, and Handan Üstündağ. And it is testing inter-observer agreement of the revised Coimbra. Can you?
Coimbra.
Okay. Method for recording enthesial changes in a skeletal sample from Kültepe, Kaniş.
Yes.
The floor is yours.
Thank you. Hello, everybody. Good morning. Thank you, the organizers and all the attenders for being here today. I will be presenting the testing inter-observer agreement of the revised Coimbra method for recording enthesial changes in a skeletal sample from Kültepe Kaniş site. This was a work conducted by a group from me and Handan Hoca and our colleagues. Activity-related changes in archaeological skeletons. It is already known that repetitive physical activity is in everyday life related to labor or subsistence activities can lead to changes in human skeletons. These changes appear as muscle-to-bone attachment areas changes. They appear as changes in structural of the bone shaft, as also as degenerative changes in the joints. The subjects that bioarcheological research concerned with activity are past activity patterns, division of labor, including gender, also status and uh, population stratifications, social status, also the specific activities and occupations. Anthesial changes is a consensual term used for musculoskeletal stress markers, also at the same time for the enthesopathies. These changes occur in the muscle, tendon, or ligament insertion areas onto the bones, and they are of they appear in the two type of entheses, fibrous and fibrocartilaginous. The anthesial changes appear in two major forms, degenerative, both of them, osteophytic and osteolytic. The etiology is multifactorial. It increases with aging. It increases with body mass. It is male-based for sex, and it increases with mechanical stress. Few methods have been developed to record enthesial changes, and the one that it's concern our research is one of the latest developed by Henderson and his colleagues, called the Coimbra method. The Coimbra method first was initiated in a workshop in 2019 in Coimbra, Portugal, and was preliminarily published in 2013. A revision of the method was conducted by Henderson and his colleagues in 2016 due to the high intra and inter-observer error in the original version of this method. This revision refined somehow the method, revised and added some of the features, and reduced the inter-observer error. The authors of the method stress 2 points: that the photographs in the publications are insufficient, and they recommend in-person training with themselves. The aim of our research, as I previously mentioned, the authors of this method stress the fact that the guidelines, and especially the photographs in the publications, are insufficient. And us as researchers looking for a standard method to assess the anthesial changes, and without the possibility to get this in-person training from the authors, we wanted to test the new Coimbra method for the inter-observer agreement using just the guidelines published by the authors of the method. The material that we used for this research comes from Kültepe-Kanes archaeological site. As you know, Kültepe-Kanes is located in central Anatolia near the modern Kayseri. The site consists of a big mound, as you can see in the picture. The big mound is called Kanes, and also the lower city, which is called Karum. The settlement has a very long time of settlement from the early Bronze Age and to the Roman period. And its importance increased exponentially during the early and middle Bronze Ages, and it was one of the most important centers during the Assyrian trade colonies during the Anatolia during the first half of the second millennium BC. The material that we used for this research comes from both Karum and the mound. It consists of 52 adult individuals, which is made of also all of the adult age groups and represented by both genders. The method that we used for this research was 4 observers recording the anthesial changes. This process took place in Anadolu University Anthropology Laboratory. And to test the inter-observer agreement, we chose 3 fibrocartilaginous entheses of the arm, the subscapularis, common extensor, and biceps brachii. We chose these 3 specific entheses in order to compare our results with previously done testing of this method. The Coimbra method divides the anthesis into two zones. The first zone is the outside border, and in the pictures is marked with a black line. This zone is the outside border, is the opposite sharp end of the fiber bone attachment. And the second zone is the attachment site of the fibrocartilaginous articular surface. And in the photograph, you can see it as marked with gray area. The features that are recorded in the Coimbra method are 6. 2 of them are recorded just in zone 1, and 4 others, together with the 2 that are recorded in the zone 1, are recorded for the zone 2. Unfortunately, I don't have a pointer, but you can see from the colorings. The bone formation appears as exostosis and anthesophytes. The erosion appears as irregular depressions. The textural change appears as a non-uniform granular surface. The fine porosity appears as cavities less than 1 millimeter wide. The macroporosity are pores larger than 1 millimeter with an unclear base, and cavitations, which are large pores with a distinct base. For this research, for the statistical analysis, we used SPSS version 26.0, and for the interobserver data analysis, we used 2 ways. The phase kappa analysis, this is an analysis advised for testing the interobserver agreement for more than 2 observers, and the percentage of agreement. This analysis, the percentage of agreement, it is by the statisticians, it is not advised to be used for such analysis, but we used it in order to compare our results with the previously done studies. The phase kappa analysis result that we got showed a wide range of results. It was a little bit consistent in the zone beer. It showed consistency in zone 1. It was high, relatively high. It was substantial and almost perfect results, especially for the bone formation features. Otherwise, in the zone 2, the results were highly inconsistent for many of the features in all the 3 antheses. In this, from the phase kappa results, we got high values for the rarely observed features, such as the cavitations. And this is also pointed by the authors of the method. Our phase kappa analysis results, it is similar to the previously done testing by Wilczak and his colleagues, the Coimbra B testing. The second analysis was the percentage of agreement. For the percentage of agreement, we got relatively high results. This was not a really surprise for us because, as I previously mentioned, the statisticians, they indicate that this increases the agreement between observers. As you can see, all the results almost are above 90, and just for 2 of the results we got 78 and 85%. Regarding this analysis, we were highly compatible with among each other, and compared to the previously done testing of the method, our results showed high differences for each feature in all the antheses. The phase kappa analysis, as I mentioned from the results, showed a wide range from slight to perfect agreement, and it shows similar results to the work done by Wilczak and his colleagues. The percentage of agreement, as I stressed a few times, showed considerably high results, and this is pointed out by the statisticians that this is not a well-fit analysis to be used for such analysis. The highest agreement for the place kappa in all the antheses was for the bone formation feature in zone 1, and it was with a big difference for all the features in the zone 2. The features in zone 2 showed only the rarely observed features like cavitations showed high agreement in our results. And to conclude, the new version of the Coimbra method may have refined it and slightly increased the agreement between observers. However, testing it in a Bronze Age population from Anatolia showed that the features, especially the ones in Zone 2, still remain uncertain and may be open for further discussions. We suggest that further simplification and unification of features may make the method more applicable, and a more detailed and clearer visual guideline seems to be needed. The moderate agreement results that we got makes the application of this method difficult to be used by researchers, especially the ones that do not have the opportunity to get the in-person training by the authors, as they suggest. And it also, this kind of results makes it hard to be used as to be considered to be used as a standard method to record anthesial changes. And this research was done in the scope, within the scope of the multidisciplinary analysis of the biological and social structure of Kültepe-Kanes population, supported by TÜBİTAK. Thank you for listening, and if you have any question, I can answer.
Thank you very much for this very, very interesting study. Are there any questions? Anything you want to ask, to learn? Why don't you ask questions?
Oh,
I don't want to be the worst chairperson, but I do have a question.
Maybe I can say something.
Yes.
I'm not yet a doctor.
I'm sorry. Okay, soon you're going to be one, I guess.
I hope.
Okay, I would like to ask the question either to you or to the doctor who is sitting in front of me. I assume that you eliminate the parameter of age, right?
No.
Because you mentioned that certainly the age is a parameter. Are you eliminating this parameter in your studies?
In this research, we didn't eliminate it because this was just the testing of the method. But for regarding the evaluation of anthesial changes regarding the activity, yes, we do, because especially the old adults, they show higher values for the anthesial changes, much more than the young adult or middle adult. So the old adult age group of the population is eliminated from the evaluation of the activity patterns from this method.
So you're almost ready to become a doctor. Thank you.
Thank you.
So let's go. Very nice. And everybody is very good in timing. Thank you for this too. I'm the one that I'm not good, very, very good at that. Now, our third speaker is going to talk about bioarcheology, the rescue site, stable isotope analysis of İYYU necropolis. Thank you. And the floor is yours.
Thank you very much. Good morning, everyone, and thank you for accepting me to this conference. It's my first real conference, not an online one, and an adult one. And for being here today, both in person and online. I would first like to acknowledge those who made this research possible. Besides the grants you see on the slide, I would like to thank my thesis advisors, Professor Rana Özbal, who is here with us today, and Professor Inge Autoruben of Koç University, and express my utmost gratitude to the head of the Juliopolis Research Project, Professor Alimetin Büyükkarakaya of Hacettepe University, who I hope is watching us on YouTube. Alimetin Hoca provided the samples, helped to obtain the permit to study them, and perform the histological analysis. I would also like to thank Professor Kamiray Özdemir of Hacettepe University, who is with us today, for his comments and suggestions related to my data as my jury member, and Dr. Benjamin Irwin, who's also here with us, for helping me with collagen extraction protocols and data visualization, and Mohamed Dolmuş for providing digitized archival records. I will give a brief overview of the site and the research project it's a part of, and will then focus on the dietary data obtained using stable isotope analysis. I refer to the site by its Greek name, Juliopolis, while the modernized version of the name Juliopolis is retained where it's officially used in the project titles. So Juliopolis is located northwest of Ankara at the confluence of the Aladağ Çayı, ancient Skopas, and the Sakarya, ancient Sangarios rivers. It was flooded in 1954 during the construction of the Sarıyer Dam. So this is the area that now remains above the surface level, which has been the subject of ongoing rescue excavations by the Anatolian Civilizations Museum in Ankara since 2009. The ongoing campaign aims to gather more data about the site and to prevent further damage inflicted by looting, which followed the first rescue campaign of 1991. Prior to that, Juliopolis was studied mainly in the scope of regional surveys. So the importance of the site, defined in the primary sources as a small town, was mainly linked to its position on the border of two provinces, Bithynia and Galatia, on an important highway used for military campaigns and pilgrimages, known as the Pilgrim's Road, that connected Constantinople to Ankara and went further to the Levant. The proximity to Ankara had to have a considerable impact on the life at the settlement, since it was a prominent city in Asia Minor for many centuries, from the reign of Diocletian until the Seljuk conquest, and functioned as a military base and a trade center because of its position as a junction point that you can see on the slide. Juliopolis was well connected to the rest of Bithynia, as attested by the coins found at the site. So the coins are from Nicaea, Nicomedia, Prusios at Hippium, and Cratea, as well as Amastris in the neighboring Paphlagonia. The microregion of the site was equally dynamic. Here you can see the results of a survey carried out by Professor Peter Brown and his team, who studied Western Galatia, the home of Saint Theodore of Sicyon, in the late 90s, early 2000s. The extent and the layout of the settlement itself are currently unclear, except for its two necropolis on each side of the Aladağ Çayı, so the West Necropolis and the East Necropolis, a circuit wall, and a church. Overall, the archaeological information available about this provincial town is limited. Therefore, its main accessible feature, a burial ground with 764 graves excavated as of December 2021, presents the most research interest. The identification of the necropolis as a burial ground was done based on the coins found there and David French's research on the Pilgrim's Road. Based on the archaeological evidence and written sources, it's estimated that the settlement was inhabited between the 4th century BCE and the 11th century CE, and the first three radiocarbon dates obtained in the scope of the Gabon grant fit this estimate, showing that the necropolis was in use at least between the 2nd century BCE and the 6th-7th century CE. The burial ground provides extensive information about the funerary customs in the region during the Hellenistic, Roman, and Byzantine periods. The most commonly used practice was inhumation, although several cremation graves were also excavated, and one of them is this Laginos burial on the slide. So the largest group of the excavated burials are cist graves, a feature which is accommodated by the geology of the area. Some are placed independently, while others are surrounded by two semicircular structures. So the other two main categories are simple inhumations and chamber tombs, with only two examples of sarcophagi placed at the highest elevation point and one cenotaph. The graves of Heliopolis have produced an extensive burial assemblage with over 800 items in the museum's exhibit and 3,000 in its research inventory. These include metal, glass, bone, and ceramic objects, precious and semi-precious jewelry, amulets, and coins. Calcium in the local bedrock contributed to the preservation of perishable materials, namely wood and leather. So on the one hand, these objects provide general information about the settlement, such as the presence of a local ceramic workshop, and represent an iconographic repertoire that can be linked to the cults practiced by the city inhabitants. On the other hand, the archaeological context on its own is not sufficient to gather personal information about the local inhabitants and their socioeconomic structures. So luckily, because of the scale and the importance of the site for regional studies, its materials were included in the Heliopolis Anthropological Research Project. It's a multidisciplinary initiative focused on bioarcheological methods led by Professor İYYU and his team since 2017 with the support of the Koç University Vehbi Koç Ankara Studies Research Center. And on the slide, you can see the completed and ongoing projects conducted in the scope of the initiative. So it has great potential for raising the bar of the research at rescue sites in Turkey, as it involves advanced scientific techniques and an interdisciplinary team, which are usually prerogatives of larger scale projects and monumental sites. And it also uses bioarcheological data to expand the line of inquiry from populations in general to individual people, their health, well-being, and life histories. So this is also the focus of the current study carried out in the scope of the initiative and my recently defended master's thesis. Access to food resources can act as a proxy of personal status and well-being and provide insights about socioeconomic dynamics within the settlement and the role the anthropological factors such as biological sex and age played in them. When it comes to subsistence strategies at Heliopolis, we know from Galin that the town, together with 4 other cities in the coldest parts of Bithynia, marked on the map, as well as Dorylaeum in the neighboring Phrygia, grew diopyros, a grain suitable for harsh climates and used to make bread. The location of the site at the confluence of 2 rivers likely provided additional opportunities for gathering food, irrigation, production, and communication, particularly considering that the Sangarius was a navigable route. The proximity to the rivers might have been the decisive factor for establishing a settlement here in an area with fertile soil, the importance of which could be linked to a local cult of Dionysus Kalikarpos, the epithet that means bearing good fruit. And this is the only information available about the subsistence at the site, as no archaeobotanical or archaeological examination could be performed. Given the availability of skeletal material from diverse burial contexts, the site presents a good opportunity for the diachronic examination of the local dietary habits through carbon and nitrogen stable isotope analysis, which is currently the best method for extracting personal dietary data and comparing it among local populations and with inhabitants at other sites. My initial project was to examine 42 individuals from the East Necropolis, a better explored part of the site, in order to interpret the nature of their diet and examine possible differences based on their anthropological and archaeological characteristics. While the necropolis has been in use since the Hellenistic period, the samples in my study come from the Roman Imperial, late Antique, and early Byzantine contexts. So here you see an overview of my project plan. The collagen extraction, based on the modification of the original logging method, preferable for poorly preserved bones, was carried out at the Koç University Archaeology Lab with the help of Professor Özbal. They were further processed at the university's Kuytam Lab and shipped to the Tenmak Lab in Ankara for isotope ratio mass spectrometry. So once the results were obtained, I assessed their collagen quality using standard quality controls in order to proceed with the analysis and interpretation. So my hypothesis was that the average isotope ratios from the site would be consistent with those published for other Roman and Byzantine settlements in Anatolia and would reflect the diet dominated by C3 plants and terrestrial animals. Since women and subadults in the Roman society are known to have had more frugal diets than the adult males, I anticipated that the local signatures might reflect that. I also expected to see a number of dietary outliers due to socioeconomic differences and high human mobility at the site. And the main challenge of the study was the poor preservation of the skeletal remains caused by the geological characteristics of the site. On the one hand, it decreased the number of individuals for whom osteological data could be obtained, and also a compromised collagen preservation. So at the initial stage, we expected that 25 to 30% of the samples would not meet quality criteria, meaning that the collagen would not be sufficient in terms of amount and quality. In reality, so this is the final data set, only 8 of 42 samples met the standards. This is comparable with the situation at Hierapolis, where 7 out of 53 samples passed the criteria. And while the initial sample set contained different types of bone, the final one that you see here consists of ribs only and reflects a diet during the last 2 to 5 years of a person's life. And bone collagen, selected based on the standard quality criteria and better chances of preservation, is formed by dietary proteins. So here we're looking at protein sources in the diet. The anthropological data consists of age at death and sex, which in some cases could not be determined because of the preservation issues. And due to this small sample set, the archaeological context was narrowed down to grave types and the number of grave gifts per individual. So the results are as follows. And the averages, the average is here. They're consistent with a diet based on terrestrial animals and C3 plants, with a slight indication of direct or indirect consumption of C4 plants, because we take minus 19 per mil as the threshold value for diet strictly based on C3 plants. They are overall similar to the averages obtained from other contemporary sites in Asia Minor, although the average carbon value is slightly more positive and the average nitrogen value is lower than at the majority of other sites. So the current results, they reflect the diets of only 8 individuals aged between 15 and 45, with children and older adults not represented. And grave types are limited to seized graves and chamber tombs. However, the small sample size still allows for some preliminary observations that can be further explored by increasing the number of samples and possibly rerunning the samples that did not meet the quality criteria by a small amount with the additional ultrafiltration step, which can improve collagen yields. I detected 2 outliers. So the individual with the most negative and the lowest nitrogen signatures in the group, over there, the furthest from me. It was a young adult female from burial 164, and she relied on C3 plants in her diet more heavily than the rest of the group and had more limited access to animal proteins. Another outlier is an adult male from burial 116 with the most positive carbon and the highest nitrogen signatures, which tells us that this person's values are consistent with the presence of C4 plants in their diet and better access to animal proteins. It's hard to give more specific information without archaeobotanical or zooarchaeological data and establishing local isotopic baselines. Overall, the 2 males in the group have the highest nitrogen and the most positive carbon values, while the 2 females exhibit the lowest and the most negative ones. Individuals whose sex could not be precisely determined occupied intermediate positions. When it comes to age, adults exhibit higher nitrogen and more positive carbon signatures, and the fact that the 2 adults in the data set are also the only 2 males identified makes it hard to distinguish which of the 2 factors, age or sex, had more weight in the heightened carbon and nitrogen values. The burial type seems to have no significant difference when it comes to nitrogen, while the carbon values seem to be slightly more positive for the individuals from seized graves compared to those in chamber tombs. And the final sample set is limited in terms of burial gift variation, so we don't have graves without any gifts in the data set. However, it's clear that the number of gifts does not necessarily indicate a better diet, since the adult male who was characterized by more protein was buried with a single bronze coin, and the young adult female whose diet appears more modest had a more extensive funerary assemblage. Overall, there are indications of possible sex and age-based differences in diet, which remain to be confirmed in the future if further isotopic data is collected from the site. This preliminary result fits well with information from the written sources about the more simple nature of diets in women and subadults in Roman society, and dietary differences observed in other parts of the Roman and Byzantine empires. Despite the limitations, the study has generated information about the lifestyles of the Heliopolis population that, in the absence of the main settlement and its floral and faunal records, would otherwise be inaccessible. The isotopic data will be stored in the digital archive of VECAM to provide other scholars with reference. It is a contribution to the body of isotopic research conducted at historical sites in Turkey and demonstrates that the country has sufficient facilities to perform stable isotopic analysis. Most importantly, this project exemplifies the benefits of complementing funerary data with the information about once living individuals that can be obtained by bioarcheological methods, as well as using these methods to explore the materials collected at rescue sites. And thank you for your attention.
Wonderful presentation. Thank you.
Thank you.
Perfect as the first presentation of yours. Are there any questions? There are no questions again? I have one. This is, you may not know the answer, but anyway, how do you find out knowledge about or data about the gender?
Okay, so through osteological analysis.
It's osteological,
yeah.
It's not DNA type.
No, but it would be
Why?
great.
Why? Okay.
Money?
Thank you, and I'm enthusiastic with the way that you collaborate all the universities. This is surprising. This is very nice, and thank you for the presentation.
Thank you for inviting me.
Well, the last talk of this section will be given by Professor Rana Özbal. It's a group of people, a researchers group that most probably is going to be seen. Yes, right now we see it. The topic is carbon and nitrogen isotope results for two Byzantine period sites in West Anatolia, Barçınhöyük and Kadıkalesi.
Thank you very much. Thank you for the introduction. So I'd like to here report the stable isotope values for carbon and nitrogen for these two sites. And comparing them across different regions is a notoriously difficult task because there's lots of differences in the local isoscapes. However, I'm trying to combine the data for the human remains with the animal remains in order to be able to understand this. And the fact that I'm dealing with two different Byzantine sites with different natures allows me to be able to compare these two sites with regards to diet and the general food preferences that were selected by the inhabitants of these sites. And so both of these sites discussed come from Western Anatolia. So how do I go like this? Oh, this did work. Come from Western Anatolia, and Barçınhöyük in the northwest Anatolia is a middle Byzantine site. We only excavated the cemetery, and Kadıkalesi is a large fortified settlement with a monumental church dating to the late Byzantine period. I will start with showing you the location of Kadıkalesi. This site is as I said, it's a large fortified settlement. It was potentially the home to members of important noble Byzantine families. The graves discovered were also associated with a monumental church, and some of them may have been members of clergy. You can see here the location of the settlement. And some of the finds. You can see that it's directly very close to the sea in Kuşadası, and this is an important indication as to whether marine resources were also available in the diet. You can see the fortifications here as well. We know that it was a port city. There's a wealth of finds, including ceramics, ivory, metal objects, glass, coins. And in fact, it was promoted, evidence suggests that this site was promoted not just to be a commercial port, but an actual customs station in the Byzantine period. So we're talking about what you can name a small city or a port city in the Byzantine period. And we compare this with the site of Barcınhöyük, which is a tiny, I wouldn't say tiny, but it's a village settlement in the hinterland of Nicaea, Iznik. And the site probably had also a Byzantine habitation component. However, this has not been excavated. The only excavations were conducted in as you can see, it's a double mound in the eastern mound of the site, where we came upon a Byzantine cemetery on the very top. This cemetery may have been connected with a church, but no evidence for this was actually excavated. But nonetheless, there was a number of burials, and they yielded interesting results, which I'd like to share with you. You can see the excavations and the roof tile burials here in the picture, as well as some of the modest finds that were found in the settlement. We do have at least one seal suggesting that this was not a complete remote site. It was connected with larger settlements. But when you compare it with Kadıkalesi, which is a port city, it's a much more rural setting. Here's a picture of some of the burials. So just to share with you some of the basics on carbon and nitrogen analysis. While the carbon can give, based on the fractionation of the carbon due to photosynthesis, it can give you the difference between C3 and C4 plants in the diet. Nitrogen is indicative of the trophic level and can give you an indication of the amount of meat and other consumables that include sort of high protein within the diet. And it can also inform us about marine pathways as well. So the analyses were done at Koç University in our laboratory. We did the steps, all the steps of demineralization and gelatinization at the university, and therefore freeze-dried the samples over 48 hours. There's supposed to be animation, but it doesn't seem to work. It doesn't seem to work. It was supposed to go, but and thereafter, after the freeze-drying process, the samples were then weighed and analyzed in Ankara at the 10 Mach facility using two different, so 2-point calibrations with the elements, the samples. You can see the standards that you can see on the slide. So the collagen preservation was actually quite good for both sites. Of the 22 samples analyzed for Kadıkalesi, 20 of them yielded good results. This is more than 90%. For the 28 samples analyzed at Barcınhöyük, we got 22 with good collagen, falling between the carbon to nitrogen ratio of 2.9 to 3.6, suggesting sort of around 80% preservation rates. So if we look at the results, we can see that I'm going to have to sort of use this and show it to you just a second. Yeah, we can see that babies or infants are extremely high with regards to nitrogen. This has to do with the trophic level, and so an infant has an additional trophic level, usually 3 per mil more than its mother during breastfeeding. So we can see the infants here extremely high in the nitrogen axis. We can see, this was going to be also animation, so I would have been able to show you one by one the arrival of the children here in green, the women in red going down, and the men over here, as well as a number of undetermined sex individuals. This has to do with potentially preservation and other factors from the site's skeletal remains. When we look at the data in an error bar form, the infants here are again taking a large portion of this upper part of the graph. And in order to be able to see the remaining individuals better, I removed them. And you can see that there's a tight cluster between all the different individuals. The unidentified sex, the male in yellow, I don't know if you can see that there, the women slightly lower over here, and slightly more, slightly less positive with regards to the carbon, the C3 plant, and or with regards to the delta 13 value, and the children over here. So you see it's sort of a very close cluster when we look at the results. Oops. In order to look at the local isoscapes and to see how these values, how they make sense and how they can actually fit within the local signatures, we need to actually take a look at the animal remains. Unfortunately, because it was a cemetery and no settlement was excavated, we have no animal remains that we excavated from the Byzantine period that were then analyzed for isotopic values. This is why we use the Neolithic level animal remains. And you can see the humans here at 10 for the delta nitrogen 15 value, and the animals around 7. When we, just if I can go back, the Byzantine values were at 11. Using this 2022 publication by Benjamin Irvine here, we see that through time, there seems to be an increase in the delta nitrogen 15 value over time. So it's possible that with a Neolithic site representing a value of 10, which is quite high, that the values for the Byzantine period could indeed have been increased by 1 per mil and become around 11. The reason why these values are quite high potentially has to do with the dairying that this region is known for. Already in the Byzantine periods, milk and dairying was a very important component of the diet, as we know from the lipid residue analyses that we've conducted at the site. We also have Ottoman records indicating that in the Ottoman period, again, dairying continued. And even today, Bursa is a very important region for dairying. So there's no reason to assume that this did not also take place and add to the component that we can see here, the animal protein component that has significantly contributed to the data that we see here for the delta nitrogen 15 value. When we move to the site of Kadı Kalesi in Anaya, you can see, and I'm sorry that it's so small, the children are here in this tiny circle. The females are here in the larger or the medium circle, and the males reach the largest circle over here. And again, I'll show you this format in the error bar form. But one thing that's very interesting in order to sort of compare this is the spread of the data. Unlike Barcan, a small settlement with probably a very standardized diet, we seem to see a range in a port city. It only makes sense, and it shows here. The isoscapes are quite different. I'd like to point out here that we see values around 9 or 9.5. If you recall from Barcanhoek, we were looking at ranges around 11, suggesting that dietary proteins were somewhat lower here, but this has to do with the isoscape, the local isoscape of the region, and doesn't necessarily mean that people were getting less nitrogen in their diet. If I go back to the previous slide, I'd like to show there are a couple of outliers, including this gentleman over here, a male, that has extremely high nitrogen values and a more positive delta carbon 13 value. And there is also another man here that has a very low value for both. We know from the Byzantine period that fasting was an important component of the diet, meaning no milk or milk products. So it's possible that this is a member of clergy or someone who had a restricted diet, potentially on purpose. But nonetheless, what is interesting is that we see this great range. And based on the data, potentially this individual here appears to have also had some marine component within the diet. Looking overall at the data from Kadıkalesi, we see that most of the individuals are, again, despite the fact that it's right next to the sea, they're having a terrestrial C3 and animal protein-based diet. This one is where we can see, again, the children in the smaller error bar, the females and the males. And the consistent difference that we see is one which I don't actually know how to explain very well, but where the males tend to have, and this we saw also with the Juliopolis example, with the males having consistently being on this part of the axis, having consistently higher delta carbon 13 values. When we look at the local isoscapes, again, there are no Byzantine animal remains that were analyzed, but looking at Mytilene, a Byzantine site here on the coast, has a mean for sheep at 6.5, and Limantepe for around 3, let's say, for basically emmer and wheat and barley, which makes sense. So the trophic level for humans, if the sheep are at 6.5, we would add about 3 per mil, making it about 9, which fits well, which suggests that this is an isoscape difference between the two sites. When we look at the sites together in order to compare the results, again, you can see the babies taking up a large component of the top of the chart. But when we take that out and take a look at the margin individuals and compare them with those in the bottom here from Kadıkalesi, we see that there is indeed a difference that is based, as I said before, on the isoscapic values and the differences in the environmental criteria that these two sites present. One thing, however, that's really interesting is looking at the actual individuals and their diets. We can see that the coefficient, the variant of coefficient here that we see for Kadıkalesi, for the nitrogen values, are a lot more varied with the person, for example, that's fasting and the person with the marine diet contributions than for the site of İYYU, where it seems to be much more clustered. Another thing that I forgot to mention is that when we look at the carbon or the nitrogen values for men and women to compare the two sexes at each site, a t-test distribution shows no significant correlation. So we can't say that there is, in fact, any kind of difference between the sexes with regards to what they eat, although we do see this consistent change with, again, in both cases, the males, this is the unknown, but this is the males, and this is the males from the low site, and the females showing this difference. One suggestion made by Pritzi Bourbeau, who was someone that does especially Byzantine isotopic results, is that it could be that there is a small component, very small, and it can also be with fresh fish, a component that changes the isotopic ratios for potentially men. And this also changes the carbon value. And another more glaring indication could be C4 plants. What makes little sense to me is that C4 plants, because millet was taxed, millet is a C4 plant because it was taxed in the Byzantine period, that this, that it was in male diets, it makes less sense to me in terms of trying to interpret the data and understand these differences. Just to conclude, so, excuse me, I just want to get my notes. So in sum, the carbon and nitrogen stable isotope results for bone collagen for 42 analyzed samples from these two sites suggest a diet based primarily on C3 plants for both sites and terrestrial animal proteins, which holds true even for the coastal side of Kadıkalesi, with the exception of a few individuals. Our comparative evaluation of diets of the rural and urban Byzantine populations of Barcın, Höyük, and Kadıkalesi respectively fit with the Byzantine records. On food, where grain and dairy products comprise the majority of the diet. Meat is consumed rarely, and marine resources vary depending on the region. And the statistical significance between the delta nitrogen 15 values between these two sites is most probably an issue that arises from the regional isotopic baseline for each site. And the carbon 13 values could be either, as I just mentioned, marine resources or potentially C4 plants. And that concludes my talk, but I'd like to also thank GABAM, the Stavros Niarchos Foundation for Late Antique and Byzantine Studies, for funding the research that I conducted here today. And thank you for listening.
Thank you very, very much for this very interesting and informative presentation. Are there any questions? To your surprise, I have no questions. Thank you so much. Thanks a lot. And now we have a coffee break, and then we come back again at 12 o'clock, right? Yes. See you then. Thank you all.
Thank you.
Hello, all welcome to the second session of the conferences today. I'm very happy. It's a great pleasure for me to be here because the title of the conference is very impressive and saying us that science come into the existence with all field of the research, for with all relationships, intersections, all fields of the scientific researches. And for this reason, it is a very impressive title for me. And I'm very happy to be here also for this reason. And I would like to thank to the organizing committee for inviting me to be here, especially to Professor Abacı Kalfoğlu. And now I would like to invite the first speaker of the session, Adria Breu, from the Autonomous University of Barcelona. His speech is on optimizing lipid recovery rates to better understanding the adoption of drying by lactose intolerant farmers. Please, Dr. Breu. Excuse me, Breu. If I'm right now.
No worries, Sonia. Thank you very much. So here we are. This presentation has been put together by a group of researchers, and it's about optimizing the lipid recovery rates to better understand the adoption of dairying by lactose intolerant farmers. Of course, myself, who I am now doing a postdoc at Koç University, and also Sidar, Gunduzap, Pinar, Özgür, Ayla Turkikulvijik, and Rana Osval, of course, who are now part of a team which is following the steps of Professor Hadi Osval in developing and working with lipid residues in the Archeometric Center at Boğaziçi University, but also at Koç University. And the results of what I'm going to present today are the first steps of a wider project, the ANSUD project, we are trying to better understand the technologies around dairying in prehistoric Anatolia, and to start unwrapping this. I think the first thing that we need to think about is lactose intolerance, lactose persistence. As you may know, lactose is one of the main sugars in milk, and this sugar is digested through an enzyme called lactase. This, like all mammals, this is supposed to happen only during childhood. So at the moment that we grow older, our body smartly stops producing lactase, because, of course, we are not going to drink milk when we are grown-ups. We are not suckling from our mother's teeth anymore. But apparently one of these mammalian species, which are called humans, decided to continue drinking milk after childhood. And then what was an apparent useless mutation in the genome became an actual advantage. And this may be one of the first and clearest evidences of evolution driven by culture, where our choices in the cuisine, our cuisine choices, our cultural developments have modified the way our genomes behave. And these started to become really clear when the genetic analysis were done on the frequency of the persistence of lactase in modern humans. As you can see in the map here, mainly the United Kingdom and also certain areas like Saudi Arabia or Western Africa show really, really high percentages of people who nowadays can tolerate lactose beyond childhood. And there are specific reasons environmental reasons that create a really high pressure that will drive these evolutionary changes. But in general, you see that the countries who have a higher amount of population showing lactase persistence also are populations which tend to drink and consume more milk. So given this, there was clearly a big question, which is, okay, so we have a genetic situation that it's not what it should be if you look at the rest of the mammals. When did this happen? When did this start, and exactly why and how did this start, and how did this spread all across the world? So we have as archaeologists, we have a tool set to try to see when and where humans were producing and consuming milk. We of course can look at ancient DNA analysis of the 910T allele, which is the one codifying for the persistence of lactase. We've done this, and many results have been published, mainly in Europe, and show clearly that there is no lactase persistence until the Bronze Age. No, not in the Neolithic, not obviously in earlier dates where there were no actual domestic animals. And this was kind of an interesting result, because one would think that the moment you have domestic animals and the moment you have access to milk from cows, sheep, and goats, that is when the selection, the pressure, and the cuisine could be there to push this modification of the genome. But of course, genetic changes may take time. We cannot do ancient DNA analysis on all the skeletons that we find, and the skeletons that we find are only a tiny percentage of the people that were alive in prehistory. So we can assume that we only start seeing the mutation in the ancient DNA analysis when a significant percentage of the population was already showing this lactase persistence. The second type of tool that we have is looking at dental calculus for the beta-lactoglobulin protein. In this case, this is a really new advance. There's only one paper that's been thoroughly analyzing this approach, the Weiner et al. 2014, and we don't have a lot of results from Anatolia, the Levant, and northern Mesopotamia yet. So it's one way forward also. But again, it has sort of the similar limitations, which are that you can only analyze the human skeletons. So you need to have excavated them. You need to have, and this will only be a tiny percentage of the population, of course. Another type of approach is the detection of specialized kill-off patterns on the herds that were being raised. Again, that tells us about the production of milk rather than the consumption of milk. And it also has the issue that it will only show up in the record when you really are devoting your animal herds to the production of these specialized products. So you have a degree of specialization, and you are expecting to produce milk and dairy products in a high scale. So it's possible that they were consuming milk and they didn't have the kill-off patterns in their faunal records. The final tool that we use, and this is the one that we are developing at Koç and at Boğaziçi University, is the use of isotopic analysis on organic residues found in pottery. In this case, what we are hoping to achieve is we are hoping to recover a trace amount of fats that had been trapped inside the pores of the pottery. These fats would be there after the pottery had been used for cooking or for preparing or for basically eating, so after pottery use. By recovering these lipids, we then apply isotopic analysis to differentiate different types of fats. And the key thing is that there is a clear isotopic signal that is directly related with milk and dairy products. And this is because the mammary glands of the cows and sheep and goats, basically all ruminant animals, have to get an additional supplement of fat directly from the diet. But there is another part of the fat that is synthesized new from the actual following the chemical processes inside the body of these animals, and therefore you have two types of fats that have been created through different biochemical processes, and this difference between these two types of fats is basically something that you can only find in the milk. So by checking whether these isotopic differences are present in our lipid residues, we can access this knowledge about the dairying and whether dairying was present, dairy products were present in these archaeological artifacts in the pottery, and therefore know if they were consuming milk, they were processing milk, and how and when and why. Also, pottery is giving us an opportunity to go beyond the human bones, because pottery is everywhere. At the moment they started, the moment humans started using pottery, they apparently loved it. So they produce hundreds and hundreds of potsherds that they left us in the archaeological site. So it also provides us with the opportunity to see representative, maybe a representative portion of the economic activities that were being done in the site. One of the main papers to put this in motion for Anatolia was this famous paper by Evershed et al., where they found that the earliest date for milk use in the Near East, and also linked that milk use with cattle herding. And this was a very significant paper in 2008, which settled the discussion between whether dairying and dairy products were something that only became generalized in the Bronze Age during the so-called secondary product revolution, or it was something that really started in the Neolithization process in this region, this part of the world, Anatolia, northern Mesopotamia, and the Levant. It was a great paper, and it was also a great paper because of some of its shortcomings. One of those being the low preservation rates of fats in pottery. To make this clearer, basically, if you count how many pots you had to analyze before finding some fat surviving inside, only 12% of the pots analyzed by Evershed et al. Preserved fats inside the porosity of the clay. This meant destroying around 900 potsherds to achieve to find the other 100 that had good residues, which, I mean, it was necessary, but again, as archaeologists, we like to minimize the destruction of our artifacts as much as possible. The second and the third type of shortcomings were that there were chronological and geographic gaps. Of course, one study can only do so much, and for example, between Pendik and Tepecik Çiftlik, there is a huge void in 2008 that was basically empty of any data. So it was hard to understand how, as you can see here, basically the milk will show up. You look at the graph, the milk is any point that's in the lower third part of the graph, the area inside the black circle. So as you can see, the area around Pendik, Fikirtepe, around Istanbul actually, and the Marmaray region, was the one that presented the highest, really high amounts of dairy products. Whereas the rest of northern Mesopotamia and the Levant and central Anatolia also had some dairy products, but the percentage was extremely lower compared with the Marmaray region. So in trying to understand what was the case, how these two phenomena that were so completely different came to be, it was necessary to bridge this geographical gap and also expand the sampling towards the east beyond Chayonu and Tel Abyad to also better understand the rest of northern Mesopotamia and Levant. So the work that has been, the work that Hadi Oswald started since then, and also the work that we keep doing nowadays in the labs, are to get this pottery and analyze through gas chromatography, mass spectrometry, and gas chromatography coupled to isotope ratio mass spectrometry, which is a lot of letters and a lot of words, that basically allow us to recover, classify, identify, and detect the lipids. And after all these years, it has become clear that one of the main limitations and one of the challenges of this type of analysis is the fact that it takes a lot of tries. We don't know if we are going to find fats inside the clay until we do the whole analysis. So maximizing the percentage of positive results is key to make sure that we are productive, that we are optimal, and that we obtain as much data as possible from the past. At least this is an investment of time and money, and we don't want to destroy the archaeological artifact if we don't have to. And so far, we've been implementing, we've been trying to understand how lipids are preserved in the Mediterranean conditions, which are clearly different from the conditions of Northern Europe, with more colder and wet climates. And since all these methodologies were first developed by British scholars working on British archaeological materials, with time we have, through chemistry, we have better understood that the processes had to change and had to adapt and had to be different for different parts of the world. And in the case of Anatolia, we need to understand that these behave like Mediterranean preservation, and therefore we need a different type of approach than just the one that was being used for England. And this is what we have been doing in these recent months at Boğaziçi and Koç. And we are showing you today a different picture because we are starting to get nice results in much more pottery. So this is a combined work of many people. This is the work of Pinar for Tepecik Çiftlik, of course, Sidar for Sumaki, and also Ayla and other students that helped with Archeology Congress. Also, we have put here some other sites like Kendala Hejala and Terhalula, where we just go straight for the optimized methods for the Mediterranean. And what we can see here, what I'm showing you, is the preservation rate. So the amount, the percentage of pottery giving us lipids, giving us a residue of the original contents of the pot. In the case of Ashapinar, we went from 7% to 21%. In the case of Actopraklik, from 10 to 35. In the case of Sumaki, from 28 to 50. In the case of Keçiçayırı, from 50 to 60. Tepecik Çiftlik, 8 to 100%. And Kendale Hejala and Terhalula tells us only the optimized method, 75 and 50%. So in total, we went from this 30% that also Evershed was finding in their pottery, and now we have achieved this 45, on average, 45% of the pottery containing lipids. And this kind of makes sense because not all the pottery would be used for cooking or would have contained food. So we can expect that maybe 20, 30, 40, 50% of the pottery wouldn't have any residues. But also it wasn't really convincing that only 30% would have lipids. So we are now much more comfortable that these preservation rates are more consistent with the real use of the pottery vessels. So, if you have to sum it up, what is the protocol that we are following now, and what is the integral procedure that we also recommend fieldwork archaeologists to follow? Is that, well, you should treat the pottery as if the pottery was food itself, because some food may be still inside the pottery. So store the pottery films of aluminum foil like you would do with any sandwich, and put it in the freezer like you would do with any other food. When you drill the pottery to get the powder that will be used for the analysis, it's also store the powder in the freezer again as if it was food. and the extraction should be done as soon as possible to minimize the risk of oxidation and other degradation. So we don't want our food to go bad. It has an expiry date. Also, we basically changing our protocols. We are using really much more aggressive extractions with 95% sulfuric acid. It's something you want to use a fume cupboard and protective gloves and goggles and many things when you're doing it. And basically, in the end, also, we have been able to access better instruments, more modern instruments, which are much more precise and are giving us much more detail about the lipids that are found inside. So, with all these procedures, what we are hoping for is that we will be able to present in the future an isoscape, a map of northern Mesopotamia, Levant, and Anatolia, showing you the percentage, the intensity of the dairy production and consumption over several years in early prehistory. This is just what I'm showing you just now is a simulation of what we could have and what we want to have, and this we think is one of the best tools to understand how this phenomenon appeared and then spread and moved around and was renegotiated or changed all across time. So thank you very much for your attention. It's been a pleasure.
Thank you very much for your very interesting speech. It is now open to discussion or any question, any comments, please.
So once we get the isotopic data, the first thing is clearly to transform these numbers to an actual meaning name. So we check this difference between the isotopes, and when we achieve the result is usually minus 3.3 to minus 7, and that gives us the meaning that this pot contained a dairy product. So it's basically a database, a list of pottery, a list of the results, and we can start to see the percentage. So okay, we have sampled 100 shirts from this site. 70% of the shirts in this site contain a dairy signal, and 30% do not. So this is 70% of the pottery that we have sampled containing dairy products. This is like the first rough big brush approach. After this, it's also important to take into account that our signal, what we are finding inside the pottery, this fat, is the results of many, many, many uses. This is the life of the pot that we are seeing. So we need to take into account that there will be mixtures, that this is not just one product they used once for butter, and now we are finding the butter. No? And there are, so basically, the current state of the art is the use of Bayesian mixing methods. So this is an R package that where we take modern reference values for animal fats, ruminant, pig, milk from different species, and the Bayesian mixing method makes an approximation on which type of mixtures could result into the isotopic signals that we have found. This gives us a margin of error. So this becomes a distribution similar to a C14 date, but instead of the axis being time, is the amount of milk. And then with these statistical distributions, you can combine them, and you can obtain the probability distribution for the assemblage that you have analyzed. In this case, instead of just being 70% milk, you would have a probability distribution for the site, maybe 70% plus minus 20. And this gives you, this is more like fine brush, and more taking into account the limitations and the variability that comes with the type of analysis that we are doing. Hopefully this is a good answer. Yes, we are focusing on milk because there is this significant historic question to answer. But when we do the analysis, we can see, of course, many other products. We can see fat from the subcutaneous part of the body. So basically, fat that would come with the meat. We can also see plant material. We can see waxes. We can see resins. We can see a range of products. It's not all the products, but a range of products that would be preserved through time and will not be dissolved in water or affected by the degradation of organic matter.
Okay, thank you very much.
I think
Is there any other questions? Just one, please. No.
so. Of course, usually when we analyze storage vessels, so big, thick-walled, very big vessels, those are the ones giving us less amounts of fat because you would not store and you would store maybe grain, you would store other types of products in these big vessels that will not give you a lipid signal. Medium to small vessels, mainly used for cooking and for eating, are the ones giving us better preservation. Also, inside the vessel, if you are boiling, if you are doing certain types of cooking practices, the fats will tend to be in the upper side because, as you know, the fat in the chorba it's floating. But if you're cooking in a different way, then the base of the pottery is the one which has been in contact with the lipids. So there's here also the effect of the cooking techniques that takes. So if you have pottery where you see indirect evidences of cooking, that's a good candidate, a good vessel to check.
Okay, thank you very much. I'm pleased to welcome Hüseyin Güdücüoğlu as the second speaker. His talk is on investigation of nutrition and disease conditions with molecular bacterial analysis in dental calculus samples.
Thank you. Thank you, Chair. Thank you. She's my colleague, actually, and she's microbiology, I know. It's for... I will talk you about my study. It is the unique in the Turkey, and this is the now will be published in September about paleomicrobiology. We are doing this congress very beautiful place thanks to organization committee, and this is very old place, and coming from the Ottoman. And my study about investigation of nutrition and disease condition with molecular bacteria analyzed in dental calculus sample from 19th century. This is my group, and they are archaeologists and dentists, and they are working in the museum in the one. Tepeköy between Muş and Barto Mount cemetery in the 19th century. As a result of excavation, some skeletons were found in this area, and 4 skulls were brought to our laboratory, and we evacuated dental calculus, only one. After this dental calculus, we start to UV irradiation of the dental calculus obtained from these skulls. Bleach first, we put to bleach, was used as decontamination for the cleaning of the other surface, and UV. And we worked with nanopore technology. This technology very new and coming from England, and DNA analyzing method. The sequencing takes place changing the current value according to the size and shape of the bases passing through the pores. First, you are doing the library process. after taking to process, working with the long read method, Oxford Nanopore technology sequence full-length fragments, so users can access read up to 100 kilobyte in length after you are doing the analyze. After you must choose some primer. Primer pair targets a region of approximately 1,500 bus pair scanning, and between V1 and V9 region of the 16S RNA gene. You are choosing this primer. And we done bioinformatic analyzing and detailed, you are seeing the detailed graphic for the analyzed reports of the samples are available in the image, and we continue based off the branch, class, team, family, and genes. We isolated and prominent microorganisms, Firmicutes in the foreground, Bacteroidetes, Actinobacteria, Fusobacteria, Protobacteria, Verrucomicrobia, Cyanobacteria. This bacteria, Firmicutes and Bacteroidetes, emerge as dominant strain. We constituting 98% of all 16 sRNA sequence in each mammalian host. We find a high Firmicutes bacteroidetes is also alters the production of short chain fatty acids. Acetate production show us increases if we find increase, and butyrate production decreases. High level of acetate cause insulin resistance and increase the production of ghrelin, appetite stimulating hormone in the stomach. Low butyrate levels in the gut promote low level inflammation that evokes insulin resistance. For this reason, these two microorganisms have been held responsible for this dental calculus person can be diabetes and obesity. We find on the PubMed some articles. The main factor causing the development of subchondral osteophytes is showing us obesity. It has been found in Buddha skeletons. This is coming from the anthropology sections. And at the class level, if you see this class level, the first three dominant microorganisms we find Clostridia, Beta-Proteobacteria, and Bacilli were detected. Actinobacteria, Acidobacteria, Bacteroidetes, Chlorophyx, and Proteobacteria were the most common group of microorganisms in rice, clover, and rice wheat cultivated soils. After all, these microorganisms are common in habits of agriculture soil. It turns out that person whose DNA is isolated was fed with rice and wheat-based agriculture products. Two bacteria we obtained at the family level, Ruminococcus burkholderia, especially in some metagenomic studies, showed that the good microbiota such as Ruminococcus, Cocci, Parabacteroides, Disassonis, and Provotella are rich in women with gestational diabetes. This can be shown as another proof that the individual in the study has diabetes. Ralstonia burkholderia is a microorganism commonly found in the environment and in many cases isolated from patients with cystic fibrosis. And conclusion, according to anthropologists sending this dental calculus information, we know that the person is between the age 20 and 13. Based on our findings, we can suggest that the person may have diabetes, obesity, cystic fibrosis, and perhaps gestational diabetes. It can be said that it's primarily fat, rice, and wheat as nutritional conditions. Maybe this is the first in the world and we are giving this, some information about the person. This is my group. Hakan Yılmaz, Serap Keskin, Tunç. Thanks. Very short and
Thank you very much. Also for short.
thank you.
Being short. Is there any
Any
question, comments? Is talk is open to discussion?
This method is giving us coverage and some bacteria passing this in this method, and this is very. Did you see this method? It's passing to bacteria level. Each time we find to these levels, and only we take to first 3 bacteria, and after we evaluate our results.
Any other? Okay, thank you very much.
Thank you.
Thank you. And now Kameray Özdemir, his talk is on stable isotope analysis and diet in the Bronze Age Kültepe Kaneş population. Please, Özdemir.
After a long time, we are
Please
together
say,
again.
Yes, I'm
We
very
will-
happy.
Yes, it's good honor for me to see you again. I hope I follow the timing because I'm famous with not to obey. So not to do. Sorry for this at the beginning. So I am going to talk about the results of isotope analysis, stable isotope analysis from the Kültepe, and possible inferences on diet. Sorry. So I am not going to talk on this graph slide because Rana Hoca in previous session explained very well about the theoretic background. So we use the stable isotopes of carbon and nitrogen. It's well established. They have well established technique and a good theoretical background. And I'm not going to talk about this as well because Donald in previous session explained the site. One thing to say, it's the important site in Anatolia. It's the biggest commercial center in the Middle Bronze Age. North Mesopotamian and Mercian established trade center. This is the biggest one. They came there, they settled, they married, so the population structure changed, they stratified. There is local people, unlocal people, foreigner, let's say different terminology we can use to describe them. And also it's important because the writing entered Anatolia. It's the first document about trade relations, many cuneiform tablets mention about this. So we want to learn more about the population structure. Handan Hoca from Anadolu University organized a TÜBİTAK, really good TÜBİTAK project, and part of this is stable isotopes to get clues for this about diet for this population. So our aim for to study to determine the dietary habits of the Kültepe Kaneş population, then to explain any difference in dietary habits according to characteristics such as burial location, time period, grave, and burial types. Then, of course, changing variation in diet relative to the socioeconomic status. For this, we choose samples. We have 65 individuals. We get rib samples from them, and 7 animal samples, 65 from human, 7 from the animals. Unfortunately, 3 samples from human didn't work, and 1 sample from animal didn't work as well. This first data set processed and analyzed in TÜBİTAK MAN in Gebze by Dr. Turhan Doğan. He used IRMS for analyze. And another data set studied by Handan Hoca consists 25 individuals. This independent study, unpublished study, she used also IRMS in her study, and we decided to use together these 2 groups to have many individuals, and then the statistical analysis say many things, and we can trust them as well. And the data set, I have variables, period and location, main variables, then burial types, then grave types, then sub variables, as there are sub variables as well. This is the number of individuals for each sample. In general, in this kind of study, we plot all the individuals in carbon nitrogen chart, scatter plot. You see they cluster together. There are 4 outliers. We suspect about their quality criteria. We decide to reanalyze these samples. So they range in small scale. The carbon values 19.6 per mil to 18.3 per mil. Then nitrogen values range between the 11.6 per mil to 8.7. Difference is small. So at first, I mean, because they are clustered together, one can say, oh, there's it together. Same thing, they share the same culture, dietary habits. So what is the reason to study? Just saying they're all same. But of course, there are some secrets behind this. And then we plot our adult values with local animals. As I said, 1 animal didn't produce enough collagen. So you see here dog samples. They have expected values because they are omnivore animals. Their values close to just working. Their value is close to human. And then we have ovicarpet sample, 1 here, the other here. It's, sorry for this. 1 here, 1 behind this, and the other one. You see that this 4 samples also behave differently, and they are below the local signal, meaning that they didn't eat any meat, just they graze as animals, or they included plant sources in their. So I mean, after reanalyze, after we get the result, we will think on them, and we will evaluate again. So there is this in so this working. Maybe I will do the ovicarp 2 has more positive carbon signals than the other ovicarpus sample. And saying has different management strategy. It has different management strategy, maybe pastoral, long away grazing. The others graze around the settlements. So then we compare our Kütahya results with other Anatolian early Bronze and Middle Bronze Age samples. The Anatolian is isotopical study samples stay here, and early Bronze Age, Middle Bronze Age Kütahya results. You see, they are higher than the others, and also the Middle Bronze Age. Early Bronze Age is mean close the other. Maybe we can say a transition from early Bronze Age to the data reflect a transition from early Bronze Age to Middle Bronze Age in dietary pattern. There's a transition. They reflect there's a transition. And we compare the Kütahya results with in Near Great Near East. Kütahya results stay here and cluster together with the Caucasian isotopic mean together here. Yes, that's scatter plot what tell, but I want to get more information, more data, more knowledge. Then I need to use statistics. At the site, there are three different grave types, namely simple illumination, then stone schist, and jar grave type. And I check difference by ANOVA. Although ANOVA attained stone schist and simple information into the different group for both stable isotopes, it's not statistically significant. Then, according to the burial type, there is a differentiation because some graves we get one individual. In some graves, we get multiple individuals. So we check it, and we see there is statistically a significant relation in carbon values. Then, of course, the location. There is a Tepe and Karum, upper city, lower city in different naming. So we check them. There is no statistically significant relation between these two groups. Then time periods, of course, early Bronze Age, Middle Bronze Age. We check by t-test, previous one also the t-test. There is no significant relation there. So at this point, I disappointed because they cluster together in scatter plot. Most of the comparisons didn't yield statistically significant relations. Just one, the burial type, single, and there is a statistically significant relation between single and multiple one. So but I know that based on the archaeological data, the other anthropological data and systems analysis, it stratifies society, and there are foreigner, local people. So what can I do? Then I decide to do principal component analysis. No, we decide to do principal component analysis. This is unsupervised result of principal component analysis. You see, this is first principal component component, and this one the second principal component. Totally, principal component analysis explain 62% variation in my data. It's a good number. Generally, researchers do their analysis based on 40%, 50%. This is 62 is a good percent, and generally accepted, trusted about the 6% result percentage in research. So, sorry, just initially the macroscopic examination think there is a group behave from the rest of the population. There's another group, we can say, and there's another one. I'm just at first I can say there is grouping. And 3. Then why not cluster analysis? We can add because cluster analysis cluster the data based on the things. This is actually PCA mix using all quantitative and qualitative data. I have carbon and nitrogen data, and also the gray type, time period, then location, differentiation, then I said period as well. So PCA mix put all the things together, it work on them, and produce some. Then based on the PCA, we did cluster analysis. You see there is some clustering. Yeah. Above this line, the cluster, the groups above this line are significant grouping. So 1, 2, 3 group. Actually, my data we can reflects 3 groups characteristics. So these are the sample number, so denote individuals. So I decide to check why they are there. Because then we studied them, and then below all early Bronze Age individuals. Then they are from Tepe, mainly simple humiliation and single burial, the characteristic of grouping. Then when check the green individuals, they are from Middle Age, Middle Bronze Age. They are from Karun, only one individual from Tepe. Then all simple inhumation, then single or multiple burial. There's also jar gray type as well. Then the red one from Karun. They are also from the Middle Bronze Age, and mainly in stone schist. There is jar burial also, but mainly in stone schist, gray type. So then I decide to color. Oh, is this not working? Sorry. I think something happened when color these individuals. It's maybe in following things. So no, something change. Mhm. Okay. So when we color this, they are blue. This part green, and this part all red individuals grouped together. Yes. Then I wonder what is behind this, because I have a group of variables, quantitative and qualitative variables together. So when I look at the loaded graph, I see there is behind this grouping, there is carbon, nitrogen, dominant, carbon, they move together, and the location, period, burial type, and grade type move together. Then I check the square cosine result. The first principal component scatter my data based mainly regarding periods, location, burial type, grade, and grade type. So on, let's go back. On this first component, the variation of my data according to quantitative data. Then the second principal components say it scattered the data, mainly nitrogen and then carbon. So scattering of data on F2 line, mainly the value of carbon and nitrogen value of individuals. So, but how still I wonder how carbon and nitrogen affects this grouping, and are there any secret information behind? So I check when I used this graph together. I mean, in its square loaded, the position of the nitrogen almost right angle, have right angle, close right angle, but almost. Then an hypothetic line I added. You see this nitrogen line. I added the graphs. When I check the values, upper part, towards the upper part, nitrogen level became smaller, and towards the lower part, nitrogen level became higher, close. I mean, let's say upper part close to 8, lower part close to 11 per mil. So when I take projection of each data with right angle, I transfer all data to on this line. I see the first these groups behave differently. At here, just mix with the red one, and then I move. Then at this part, these two groups nitrogen values mix each other. So we can say this group is the lower or they have lower protein intake in their diet, and these two groups have a higher protein level in their diet. So they actually, these are the upper group is rich, poor, lower groups are the richest, and these are of course the top bottom on the bottom, the highest or the richest concerning the diet. Richest people stay there. So I did same. Yes, well, so I did the same things for the carbon. Since carbon situated in graphics at with 45 degree angle, I added carbon graphs, hypothetical carbon line, this one, and towards the upper part, carbon values more negative, let's say minus 20. Towards the lower corner, carbon values stay, shows more positive value, close to 18 per mil. So when again take the projection of this data and transfers on the line, I see this something happened again. You see? I can't see. So on this blue and the green groups share same carbon sources. It means there is something in the early Bronze Age, same animal herding practice continued in the Middle Bronze Age by that group, maybe the local group. Then there is rich one. They come with because their carbon sources differ from each other. The other two, then they use different carbon sources. So they are another group in the society. Maybe they eat animals with different management, coming from the different management, animal management strategies. So my results, sorry for this. First, the results, the isotopic values are indicative of a terrestrial predominantly carbon-based mixed diet. Nitrogen value is high, means there's high in protein intake to diet. Then the majority animal protein input come from ovicarpid. Early Bronze Age sample populations, slightly more similar to the other Anatolian early Bronze Age population, may indicate transitions change between the İYYU and Middle Bronze Age in dietary habits. Kültepe, more similar to the South Caucasian population in İYYU. Maybe similar agricultural system, subsistence strategies based on the ovicarpid herding strategies. Then dietary habits more homogeneous in early Bronze Age. Single burial and simple inhumation also characterize their burial custom. In Middle Bronze Age, more heterogeneous population are there. Two different groups regarding diet and the burial custom. First group has similar carbon values as the EBA, low nitrogen values, means low protein, and mixed burial customs. And the second group characterized by different carbon values, higher nitrogen values, and multiple burial instances grave. They have this burial custom. So we can say in Middle Bronze Age, population becomes stratified. There are strangers or the unlocal people. Probably they came from the Mesopotamia, the other part of the world, because it's a trade center, and we find some clues. At the beginning, I said that because they cluster together, and the statistics say nothing, the traditional statistic, but the PCA and other hierarchical clustering say more about the status differentiation. Then I have last slide. We want to thanks to the Dr. Gondola Miller from Reading University permitting of the lab facilities used by Dr. Handan Üstündağ and Benjamin Irwin from Animate for producing scatter plots and also valuable comments on the data. And of course, the TÜBİTAK supports our project organized by Dr. Üstündağ from Anadolu University. Thank you very much. And sorry, at the beginning I said I am very famous with this. Okay. Thank you.
We have not many time, but I'm still interested in months.
Yes. No. Yeah, they came from the different, maybe different. They ate from different animals. Yeah. Yes, yes. 7 years. 7 years. Turn up. No, 7 years. I think Benjamin, it's 7 years. Hmm. Mhm. Maybe. Yeah, maybe, and possible they yield. Yes. Maybe we will include this and long bones and the rib bones together from same. Yes, it will work to check at least the results. Okay.
Okay, thank you very much.
Thank you.
Kameray. And now the last speaker is online and waiting for us. Her talk is on paleogenomic study on prehistoric individuals from the eastern Italian Alps. Please, Miriam Krause.
Thank you for your introduction, and thank you for letting me talk about my research today. So the title of my research, as you tell already, it's paleogenomic study on prehistoric individuals from the eastern Italian Alps. So why are we interested in this specific region? So on this map, you can see this region. It's in the north of Italy, and it's a border area between the Mediterranean and Northern Alps. And what is interesting in this region is you have a very complex territory with on one side isolated mountains, which means you will have isolation of groups of individuals and possible genetic differences on a regional scale. And on another side, you have very wide valleys, especially valleys going from the south to the north, and you will have movement and interaction of individuals in these valleys. And it's why this region play an important role in genetic and demographic history of European population. But indeed, you don't have any ancient genetic data available in this region, except the well-known Tyrolean Iceman that you can see on the right. And this individuals is from the Copper Age, and it has been studied a lot. And what we know about this individual, it's first, we know that he has genetic affinity with modern-day Sardinians, but also he has affinity with ancient European and Anatolian individuals from Neolithic. And indeed, why is he has affinity with this Anatolian individual? It's probably due to the migration waves that happened during Neolithic, with a migration from Anatolian farmers to Europe, and this population replaced or admixed with the local population, which were hunter-gatherer. And so the aim of your project is to indeed add and increase the genomic data available for this region in order to provide a more comprehensive picture of the genomic variation of prehistoric Europe, and at the same time from the eastern Italian Alps. And what we want to do is to determine the haplogroups of the individuals, but also we want to explore if we have any kinship in our individuals. And finally, we want to characterize the genomic diversity and the structure of these alpine prehistoric samples. So the first thing was to get samples, indeed. So we got samples from 19 sites with estimated time for the samples from Mesolithic to Iron Age. So on the right side, you have the name of each site with the number of samples. So we selected the most well-preserved sample, especially we were looking for well-preserved pars petrosa and tooth, because it's skeleton part where you will find most of the ancient DNA. And so what we did on these samples, so I forgot to tell you, in total, at the end, we had 56 samples, and on these 56 samples, we did a radiocarbon dating, and we could get data of 48 samples. And so you can see on the plot on the right, the dating of all of our samples for each site. And so you can see we have one individual from Mesolithic, then we have several individuals from Neolithic. So one bar represents one individual, and when you have more than one individual, I put in brackets the number of total individuals. So for Neolithic, we have, for example, 12 individuals. And then from Copper Age to early Iron Age, you can see we have a lot of individuals going really through all this period. And so once we have the dating, what we did, it's like we did a first molecular screening by shotgun sequencing of these samples. And the first step, it was indeed to check the quality of our samples. So first, what we did is to check human reads, which means the human endogenous content that we have. And we excluded 5 individuals with human reads inferior to 1%, because in this case, if it's less than 1%, generally you don't have enough data to do any statistical analysis next. Then, so on the plot down, you can see the percentage of human reads of all the individuals, and we basically excluded the 5 on the left. Then we check for contamination. So again, we excluded 1 individual with contamination superior to 5%. So again, we put this threshold that if you have more than 5%, then we can't know if the data that we ask come really from our sample or from the contamination. And finally, the last step for this checking was the authentication. the authenticity of the samples, which means if we really have ancient DNA, because contrary to modern DNA, in ancient DNA, you have this process called postmortem deamination, and it is what you can see in the plot. There you have the frequency of deamination by position from the 5' end of DNA, and you can see you have high frequency of deamination in the first basis, in the first position, and it's typical from ancient DNA. And all our samples show this pattern, which basically means all our samples are really ancient DNA. So now that we did this first step, we at the end have selected 50 final samples with an average coverage from 0.009 to 0.51. And so the next thing that we did with these samples is to determine the haplogroups, so mitochondrial DNA haplogroups and Y chromosome. And we also determined the sex. So we did a molecular sex assignment, and we found 24 males and 26 females, which is nice because it's almost 50-50 of each. And what you can see is on the left side, you have the Y chromosome haplogroup, and you can see you have very few haplogroups indeed that we found. With one was I2A2A1B, which is in the Mesolithic sample, which is typical from this period in Europe. Then you have, for most of the individuals, haplogroups belonging to this G branch, with then G2A, which is predominant in Neolithic European population. And then on the other side, when you look mitochondrial haplogroup, you can see that in this case, you have a very high variation in the number of haplogroups. You have in total 18 different haplogroups in our samples, with the main one is G1, K1, and H. So then after determining the haplogroup, what we did is also looking for kinship. So we used two methods, and we only kept like results when the two methods were agreed. And we found indeed several couples that are related, and they are also from the same site. And most of the couple are related to the first degree. So here you have indeed 3 individuals, and the last one is related to the second degree. And one also other interesting things is we check for the haplogroup for this individual, and we can see that you have some mitochondrial haplogroups that are similar between related individuals, which indicates a maternal relatedness of this individual. So then the next step was to do a principal component analysis. So the idea is to look what is the genetic affinity of our individual with first order modern population. So in this case, we only do the PCA with modern population from Middle East, Caucasus, and Europe. And what you can see is first, you have the Mesolithic individual on the left that is clearly different from any modern population. And on the other side, most of our sample cluster together with the Sardinian modern population, which indeed it's what has been found with the Iceman, which is also from this region. And you see some individuals that are from Bronze Age that's a little bit differentiated, and especially one that is clustering with West modern European population. So then the next step, it was to look for ancient individuals. So what we did is we project ancient individuals from Mesolithic to Iron Age on the modern PCA. And what you see here is first in blue. If you remember, it's where we have our Mesolithic individuals. We were alone before, but now you can see it's grouped with hunter-gatherer from Europe, and indeed mainly with a Western European hunter-gatherer. Then you have a second group where you have this blue and green color, where it's mainly Neolithic and Copper Age individuals from Europe, and indeed it's where most of our individuals are. And there I also show you why the Neolithic Anatolian individuals, because as I told you before, we know that during Neolithic, you have this migration wave of Anatolian farmer from in Europe. And here you can clearly see that from Mesolithic to Neolithic, you have a changes in the genetic of the individuals, and they got a lot closer from this Neolithic Anatolian. And finally, you have a last group, as you can see in pink and purple, which is mainly Bronze Age and Iron Age individuals from Europe. And there you have one individual that were clustering with West European modern population, and some like Bronze Age individual there, down. And indeed, during this period, we know that you have a second migration wave that happened in Europe, and it's a population from Russian steppe that came during Bronze Age in Europe and mixed with the population there. And so again, we check where is this steppe population, and you can see it's on the top of the plot, and clearly you can see all the individuals from Bronze Age and later like went in the direction of this population, probably due to admixture. So the next step, it's like, okay, we see some genetic affinity with different population, and we wanted to look if we can see some like ancestral component and ancestry in our individuals. So for that, we did an admixture analysis, unsupervised one, and we use the same data set than the PCA. And there I only represented the individuals, like our samples. So each bar is one individual, and there are other from Mesolithic to Iron Age. And on the top, I put some like interesting populations that I told you, like the hunter-gatherer, the Anatolian Neolithic, and the Russian steppe, to compare with our individual. And first, what you can see, it's for the Mesolithic individual, it clearly have one main ancestral component, which is brown, and which is the same that what you find in Mesolithic hunter-gatherer. So you can clearly see that individual from Mesolithic, it's genetically similar to this Mesolithic hunter-gatherer from West Europe, and Villa Bruna, which is one individual from North Italy. And then you can see from Neolithic, you have totally different ancestral components. So from like almost all brown, we pass to mainly pink and green color. And this ancestral component, indeed, you can see that they are found in this Anatolian Neolithic population. Then you can see a small proportion of this brown, which probably due to admixture between these farmers we arrive in Europe with this hunter-gatherer. We were already there. And then you can see from Copper Age, early Bronze Age, a little bit of this beige color appearing, and it's probably coming from this Russian steppe migration. And interesting, one individual, a very high proportion of this ancestral component, and it's the one individual, if you remember in the PCA, that was really more on the top, and that was clustering with West modern European population. So in conclusion, in this project, what we show is first, like we found some close maternal and paternal relatedness of some individuals from same site. Then we also look of haplogroups, and we see that we have a high variation of mitochondrial DNA haplogroups compared to Y chromosome. Then when we look of our samples, we see that the Mesolithic individuals differ genetically from all others alpine individuals, and is genetically like Western hunter-gatherer. We clearly see evidence of this migration from Neolithic Anatolian farmer to these Eastern Italian Alps with our Neolithic and Copper Age individuals that are really genetically close to individuals from Europe from the same period, but also getting close from these Neolithic Anatolian farmers and with the same ancestral component. And finally, for the Bronze Age individuals, we see some of them that are a little bit more differentiated, and we have one that is really different and is showing more affinity with modern West European population, and it seems like he has more ancestry from this Russian steppe population. So with that, I would like to thank first Valentina Koya, which is the principal investigator of the project Genomic Diversity of Prehistoric Individuals from the Iceman's territory in the Eastern Italian Alps. So the results that I presented today are part of this project. Then I want to thank Alice Paladin, who did all the anthropological work. Then Sofia Alemano and Stefania Zingale, who did the laboratory work. And our collaborator, Annalisa Pedotti and Thorsten Gutter. And I also want to mention that all the work, laboratory and anthropological work, has been done in the İYYU Research Institute here in Bolzano. And thank you for your attention.
Thank you very much for your interesting speech. And it's open to discussion. Any question, comments, please.
I have a question. I have two questions, actually. Can you hear me?
It's better than I repeat. Thank you for this wonderful presentation. I have two questions. The one, maybe I wasn't able to clearly understand. Did you collect your samples from different areas that you showed us in this map in the first few slides?
Yes, so each dot was a different site where we collected. Yes.
Then you told us that you have first degree relationship between these people that are from completely different areas at that time. Is this a question mark for you too? It was a question mark for me. Do you have an explanation for this?
So, sorry, maybe I can come back. So basically it was couple that were related from the same site, and we found different couple related. So for example, you have one couple, so two individuals were related to first degree from one site. Then the second couple was related together.
So those that were related were from the same site, right?
Exactly. Same site and same period.
Okay, thank you so much. And the second is, what did you study hypervariable regions in mitochondria?
So sorry, could you repeat?
Sorry, did you study hypervariable regions in mitochondria or something else?
So in this case, we sequence like the full mitochondrial DNA. So I look for all the SNPs.
Thank you so much. Congratulations. Any other question, comments? Yes, please.
Thank you very much. I'm from Archeology Congress. I would like to know about the region. You said about some areas.
Please, can you come here and take the microphone, please?
Hi, my name is Yavuz. I'm from Karabük University. As far as I understand that, there's a quite relation between the Anatolian DNA reports or something else, like the evidences and the foundings in the Alps. Is that true?
Sorry, can you repeat?
Okay. You said that you have some relative evidences in Anatolia in the DNA reports or something else like that. Is there any relation between these two reports but I would like to understand where was the region in Anatolia. Is that clear that can be learned, especially the specific region? Where did you find these kind of things in the Anatolia? Thank you.
So for now, the Anatolian that I use is like several region, and indeed it's one question that we want to answer later. So for now, we didn't check for specific region from Anatolia, but it's one indeed question that could be interesting to test later if they are related to a specific region from Anatolia, like more maybe central or north. But for now, I don't have the answer. Sorry.
Thank you.
Okay. We are finished the speech, and lunch is waiting for us. Please, until 2 hours, we are in the lunch break. Then see you again downstairs in the entry, and then see you again in the third session of today. Okay. Thank you.
Gracias. Dos. Gracias. Tamam. Kalem al.
Hocam sadece bir kişi sizde present. Diğerlerinin hepsi online.
Hepsi online. Tamam.
Sıkıntı yok zaten hazırladım.
Tamam.
Şu mu? Evet. Şimdi. Yok, şunu silmedim ki. Niye bu yüzünü vereceğim?
Telefonda bir şey diyemedin ya.
Sandım.
O halletti o.
Bir şey oldu.
Teşekkür ederim. Fark etmez bir abla.
Tamam. Gayet iyi.
Tamam, teşekkür ederim.
Oto şeyden dışarıdan mı başlıyoruz? Yani Atanasios şeyde mi? Online mı? Tamam. Anladım. Toplam 4 online, 1 in person. Okay. ...20 dakika değil mi sunumumuz üstünde?
15 dakika.
15 mükemmel.
15 dakikada.
Tamam, tamam.
Evet.
Dear colleagues, friends, so it's time to start session 3, part 3. I think the first presentation is going to be online. So we're going to connect to Greece, I believe, and they are going to speak over the internet. Hello, Athanasios. I'm
Hello.
Professor Karacan. Welcome to our meeting.
Hello.
You hear us, right?
Yes, can you hear me?
Okay, yes, we can hear you. Welcome to our group. So now it's time to start your presentation.
First of all, I would like to say that I'm very happy participating in this meeting. I have been the previous one in Ephesus, and Istanbul is a city that I love really. I have been several times there, and I will be also by the end of this month in another meeting in person. So I would like to thank the organizing committee and especially Professor Troboukis, who kindly had asked me to participate. Now, the title of my presentation is Dogs and Vultures, Devouring Corpses of Victims of Pandemics and Killed Enemies, an historical review. And I will discuss a bit longer the plague of Istanbul of the 6th century and the dogs in its part. Now, a big problem recurrently arising from pandemics and other great disasters is the handling of the amassed corpses. What to do with all these dead people? Everyone speaks about the work of doctors, of nurses, of politicians, but very few people speak about the people who arrange the funerals. And this is a big problem, have been a big problem since antiquity. Now, in this particular session, I will discuss the blasphemous solution given by prey dogs and vultures. To facilitate better to follow my talk, I show you a short list of the entities. So I will speak dogs in pandemics, in wars, as punishment, as a peaceful excarnation. Now, let's start with epidemics. The first notion of dogs taking part in epidemics was by Homer in Rhapsody of Iliad, and he offers the first example in Western literature. He writes, "Sing, O goddess, the anchor of Achilles, son of Peleus, that brought countless ills upon the Achaeans. Many a brave soul did it send hurrying down to Hades, and many a hero did it yield a prey to dogs and vultures." So this happened not very far away from Istanbul, in Troya. Now, in another indirect notion about the part of dogs in epidemics comes from Thucydides' famous description of the plague of Athens in the 6th century BC. He writes that, strange enough, the dogs there didn't eat the dead of the pandemics, as they usually do, because the odor prevents them to do so. The same is referred by Procopius of Caesarea on the 6th century plague, the Justinian plague in Istanbul. There were thousands, hundreds of thousands of people died. The whole city was full of dead bodies. First of all, they buried them, then put in towers, then thrown in the sea, then in big wells, dry wells, so many that there were special persons just pressing with their feet the dead corpses in the wells. But because it took several days, the pus from the previous corpses was extracting out. And then it's a very nice similarity, Procopius writes, that it's like we put grapes and press them down to take the wine, press with our feet. And the same happened then. But in Istanbul, in Justinian period, the dogs didn't eat the corpses because, again, it didn't smell well. On the contrary, John of Ephesus, from the same pandemic, 6th century in Alexandria, he writes that dogs, they were eating the dead people. And add something similar to this COVID pass we have to be able to go around the streets or hotels.
People during that pandemic in Alexandria had to carry a label, metallic label from their neck, writing their name and their relative's name, so if they die in the street of the pandemic, people can notify their relatives. But in Alexandria, the dogs did eat the dead people. Nowadays, we see something very similar with the same, with what happened then. We see a dog nipping a COVID victim still on hospital stretcher waiting for evacuation, and this comes from media. A lot of dogs were eating people in India when they died in hospitals or buried in very shallow graves. A macabre observation was made in New York, where 4,000 coronavirus victims led to vultures to cycle above Manhattan, and we can see here the vultures flying Manhattan sky. 40,000 victims were last year. Now must be, I suppose, about 80 or 100,000. And this about epidemics, but then what dogs in wars? In medieval saga, the man-eating wolf, the dog's ancestor, walks on the battlefield, and the same is true for hyena. But dogs have also depicted in protecting the master's dead body. You can see it on the left, or lamenting when he was dead. Lord Byron, in his famous poem The Siege of Corinth, in a fight between the Turks and the Venetians there in Peloponnese, writes, gorging and growling over carcass and lib. They were too busy to bark at him, the dogs. From a Tartar skull they had stripped the flesh, as he peeled the fig when its fruit is fresh, and their white tusks crunched over the white skull, as it slipped through their jaws when their edge. In more modern times, we see that after the Battle of Gettysburg in America during the Civil War, again dogs were eating the dead soldiers. After the rebellion in East Pakistan, the military forces of Western Pakistan forced millions of civilians to flee to India as refugees. On their road to the border, many lost their lives either to hardship or due to epidemic, and they were eaten by dogs. In the civil war in Angola, the dead locals similarly were eaten by dogs. And in Rwanda, after raping the captive women, the Tutsi rebels would throw them as food to the dogs. Recently, it has been reported that during the Ukrainian conflict there, dead Russians and Ukrainian soldiers were cannibalized by dogs. And then we see the part of dogs as punishment. After the execution, those convicted of various offenses, mainly against the regime, were left exposed so the vultures and dogs could feast on their bodies. The first example comes again from Homer. In Iliad Rhapsody 24, he describes, then answered Priam. Priam were going to the Greek battlefield to ask for the body of Hector. Then answered Priam, if you are indeed the squire of Achilles, son of Peleus, tell me now the whole truth. Is my son still at the ships, or has Achilles hewn him lip from lip and given him to the hounds? 4 centuries later, Sophocles returns to this topic in his tragedy Antigone. To avoid dishonoring Polynices' corpses, ordered because he had campaigned against his homeland, his sister stated, but the hapless corpse of Polynices, as rumor saith, it had been published to the town that no one shall atop him or mourn, but leave unwept, unsepulchred, a welcome store for the birds, as they espy him, to feast on at will. And the guards who were guarding the body were surprised in the morning, they couldn't see it, and no sign met the eye, as though any beast of prey or any dog had come nigh to him and torn him. Already the Leviticus sets forth a course for sinners, stating that the corpses will be eaten by dogs. Isabel then informed Ahab that he could successfully seize possession of Naboth's vineyard. Nonetheless, Eliah condemned Ahab for committing theft and murder. As punishment, God decreed Ahab's death and annihilation of his royal line. Isabel's death was also decreed, with her corpse to be devoured by dogs. And the Lord declared, I will appoint for them 4 kites of doom, the sword to slay, the dogs to drag off, and the birds of the sky and the beasts of the earth to devour and destroy. Crucifixion was often the case with convicts crucified en masse. Although it has been linked in the Christian world with Jesus' death, it was nevertheless a tolerable punishment for rebels. Indicatively, 3,000 rebels were crucified by Darius of Persia in 509 BC. Alexander the Great crucified 2,000 rebels on Tyre in 336 BC, and Alexander Iannaeus, king of Judea, crucified 800 prisoners at 886 BC. After the Spartacus defeat, 6,000 of his followers were crucified along the Appian Way in 71 BC. Caesar Augustus crucified 6,000 horsemen of his opponent Pompey in 36, while the Roman general Quintilius Varus 2,000 Jews in 4 BC. Hagic bodies were either eaten by the dogs, their lower part, or by vultures, the higher part. And when they were cannibalized, then the skeleton fell down. It is surprising from the paleoarcheology that very few remains of crucified people have been discovered with traces of dogs' teeth on their feet. Several reasons have been supposed for that, but we could expect much more to be found with so many crucifixions and so many traumas of dogs. However, the human race took its revenge by crucifying dogs themselves. This happened annually during the Supplicia Canum, its punishment of the dogs, held in Rome on the 3rd of August. During it, the dogs were suspended from a furca, a wooden fork, or a cross, and were paraded. It was based on the supposition that dogs didn't bark to alarm the Romans when the Gauls attacked the Capitoline Hill, while the geese raised a noisy alarm. Hen geese were also paraded in the same event, dressed in gold and purple. But dogs didn't devour only the dead or the battlefield. They were widely used from antiquity to hunt down and eat opponents alive. The practice was followed by the Celts, the Druids, the soldiers of Elizabeth I in Ireland, the colonies against the American Indians, and the 17th century by the Persians for the execution of convicts. All this is very alarming, battles, epidemics, punishments, but there are also peaceful excarnation of humans by dogs. And we can discuss that. There was a habit, and there is still a habit, by the Buddhists to expose the dead people, the dead bodies, on the top of hills or high raised buildings, which they call the Tower of Silence, for celestial burial. There, they leave the bodies to be cannibalized by dogs and by birds, and so only the skeleton remains, a very ecological kind of burial. But nowadays, because of the COVID epidemic in Nepal, in India, it isn't very easy to do so, because the vultures have eaten so many corpses, they don't want anymore, they don't approach the dead people. We can see here the interior of a Tower of Silence. On the outer circle are the dead bodies of men, then of women, children, and you can see the vultures sitting at the top. But there is also the ugly face of medical research. At the Body Farm, at the Forensic Anthropology Center of Texas State University, San Marcos, Texas naked bodies of dead donors are left in the fields until their flesh is fully devoured by carnivorous vultures, and various faces are recorded for research purposes. It is interesting to notice that several people, while alive, they donate their body to this farm to be left unburied and eaten by vultures for the sake of research. I cannot believe that. I think the real motive is some rejection of the society's norm or some religious feeling. Or anyway, you want to be controversial, so you leave the body to be eaten by dogs or what. Now, in conclusion, we conclude that in spite of modern sensitivities, dogs and vultures collaborated with humans in clearing those succumbed either to pandemics or to state violence. Thank you very much.
Thank you very much for your presentation, Athanasios. So we have a few minutes to discuss your presentation. It was very interesting presentation. So any questions from the hall, please? Okay, so I have one question. Actually, I was not aware of dogs playing that role in history and their names written in these literature. So in the Bible, in Homerus, so they have played very important role in the history, as you mentioned.
Yes, dog supposed to be the best friend of man, but when he dies, maybe it's worst enemy, not his master's dead body. There are very few instances recording dogs to eat their master or mistress when they found dead. Recently, we read in Greece about an old lady who died alone in her house because she was abandoned by her children and couldn't eat or what, and she died on her bed and was discovered a few days later. But half of her was eaten by her beloved dogs who were there and hungry. So dogs have a very controversial role.
Yeah, you're right. So thank you very much.
Thanks.
Thank you coming to us and giving this presentation. See you soon.
Thanks.
Now it's time for the second presentation. Is it going to be online, Soto, or in face-to-face? Online, okay. So if the speaker is ready, we can go and see them. All right, so now the second presentation is coming from Greece again. It's related to terminology of thyroid. And it's going to be presented by Chakaris. Can you hear us?
Yes, I can hear you. The name is Christos Tsagaris. Thank you very much for the invitation and for the floor. It's a great conference so far. The previous talk was really interesting, and I hope that this talk will be interesting for you as well. So the topic of this talk is the thyroid. All of us, those of us who have a medical training, have encountered in our studies the thyroid gland and its pathologies. And most of us, even those who don't have medical training, will have encountered some discussion about thyroid diseases or medication for the thyroid. What all these diverse experience have in common is that probably the main term of this thyroid is misspelled. And there are etymological and historical grounds for this, which I'm going to explain through this very presentation. So without further ado, I'll go to the next slide. And as we all know, the thyroid is an embryological offspring of the primitive pharynx. It is an endocrinological gland that is located in the anterior part of the neck, and it's wrapped around the trachea. But the question is, how are we phrasing this? The very term thyroid that we usually use, or there is another more correct term? To answer this question, we have made both a historical and an etymological analysis. Etymology is the study of the history of the words, of how they evolve, their meanings, and how they develop until today. And for the historical analysis, we went through original ancient Greek texts from the 5th century BC to the 9th century AD to see how the term evolved there as well. Let's start with the easier or more straightforward part, which is the etymology. The origin of the thyroid can be traced back to ancient Greek to the term or adjective thyroides. And this means seal-shaped or door-shaped. This is because it comes from the word thyreos, which has a double meaning. It is either an oblong door or a shaped shield, a shield like the one that warriors hold in wars. Thyreos, this word comes from thyra, and thyra means door, and it is modified into an adjective by a productive suffix of apurtenants eos. So what is important here is that we mentioned that this term has two meanings, a door and a shield, but these two meanings did not coexist for a long time. In ancient Greek texts, from Homer until the 2nd century BC, it is mainly used as a door. So when the Cyclope in the Odyssey puts a huge rock to close the entrance of his cave to Odysseus and his men, it is a thyreos there. But later on, and as the military techniques evolve and people start to use seals like the ones you can see, oblong seals like the ones you can see at the bottom of the presentation near the arrow, we have mentions of thyreos as the name of the shield. This happens in Plutarch, who is a renowned historian, and later on in the Middle Ages, as you may know, kings, warriors, knights, and warlords would put their heraldic, their emblem, their logo, if we may say, on the shield. And for this reason, thyreos also meant the emblem, the heraldic of a warlord or a king, as you can see in some examples. So at the end of the day, in the AD part of time, we have thyreos as a shield and as an emblem, and not as a door. So let's keep this in mind and move to history. From a historical point of view, ancient Greek physicians and philosophers were, of course, aware of the disorders of the thyroid gland, particularly bronchochyle and exophthalmos. This is something that Socrates, the renowned philosopher, suffered from. Aristotle and Galen knew the connection of the thyroid with the brain, so they knew the thyroid endocrinological axis, but because of the limited access to cadavers and dissections at that period of time, most of them did not distinguish the thyroid from the other glands that we can find in the oral cavity and the neck. What is more interesting starts when people are allowed more or less to dissect cadavers and make observations on them. The first one who drew the thyroid as an anatomical organ but did not name this discovery was Leonardo da Vinci. After that, Michelangelo was also assigned the task of drawing the thyroid in the De Anatomical work of Realtus Columbus. However, he left this work unfinished, so we still have a drawing of the thyroid, but not a name for this. Then Bartolomeo Stacchi provided a similar illustration, which was not published for about 2 centuries. And at the end, Andreas Vesalius was the first to describe the thyroid gland and its functions, although he also did not name it. So we have a drawing, we have a description, and yet we don't have a name of this. However, the shape of this organ that is described looked like a medieval shield, probably medieval shield with an heraldic. And for this reason, in his work Adenographia, the anatomist Thomas Wharton in the 17th century AD, based on the similarity between a coat of arms shaped shield and the thyroid gland, named it thyroid. So, thyroid and why it is called thyroid. It's probably, we don't know the exact reason, but it's probably a mistake because the person who named it and used the term was not fully aware of Greek grammatic. So thyroid is a mistaken version of the correct version thyroid. What is missing is an E, an epsilon. Why is this important? Why are we discussing this? To our knowledge, it has not been discussed in peer-reviewed literature so far, so it's something important to know. On the other hand, the correct term, of the term that we claim that it is correct, thyroid, is already used in a number of studies, about 42 in PubMed, and these studies come, for some reason, only from Eastern Europe. Perhaps the translation of the thyroid in the languages that are spoken there and the uniform system of medical education that was there in place during the last century before the demolition of the USSR helped so that this terminology is prioritized over the thyroid that we used wrongly in the rest of the world. So what we can do now with this knowledge? Of course, replacing the term would not be reasonable. Imagine having to revise all the articles we have, all the textbooks we have, and so on. However, it would be worthwhile to take some steps in order to gradually introduce or correct the term by adding this epsilon and its derivatives in the future. This can be done easily while writing articles or writing textbooks. However, we should understand that changing the term completely and leaving outside the old term will cause a problem in searching literature about thyroid. And so we need to make sure that this is something that coexists in the medical subject classifications. And in this sense, we should probably encourage the parallel and alternate use of the two terms so that gradually we have the correct term in all textbooks and articles in a number of decades or years. Certainly, we need to ensure that the adaptation of the spelling needs to be effected by degrees, gradually, and should not by any means pose any additional burdens to researchers, physicians, patients, and administrators who already face significant burden with their other tasks. So if we need to keep something from this presentation, probably it is this very slide. And what this very slide means is that we have a thera, the door, and this is thyroid, the term that we use today. This is not correct. We have thereos, the shield and the coat of arms, and the derivative adjective is thyroid, and we also have the gland. If we compare the shape of the gland with the two of them, I think that we will agree that it looks more like the second one, so the correct term is thyroid and not thyroid. Putting the E there with a parenthesis probably will be helpful. We have published short communication with these findings. If you're more interested in this, you're welcome to read this or scan the QR code there, or of course get in touch and we are happy to discuss here or later. With this short literature, and thank you very much for your attention.
Thank you very much. It's an interesting presentation about etymology. So now we have a few minutes to discuss over this issue. Any questions from the ground, please? Okay, no questions. So thank you very much, and it was very nice to have you among us. See you soon, Dr. Chakaris.
Thank you very much.
Okay, now we need to move on. So we're going to listen to third speaker today. It's going to be in our among us in the room, Dr. Mrs. İlayda from Istanbul University. Now it's your turn.
Thank you. Thank you everyone for coming here today. I'm Dr. İlayda Sham. Okay, can you hear me now? Okay, thank you. I'm Dr. İlayda Sham. I work as a resident at Istanbul University, Faculty of Medicine at the Department of Forensic Medicine. And today I'm going to be talking about the exhumation and forensic medicine and forensic anthropological examinations of skeletonized bodies and skeletal remains. But first of all, I want to talk a little bit about the forensic medicine structure in Turkey. Currently in Turkey, legal expertise services are widely carried out through the forensic medicine structuring under the supervision of Ministry of Justice. The Council of Forensic Medicine department at universities and institutes provide these services. Exhumation, postmortem examination, and forensic autopsy procedures are conducted in the presence of a prosecutor or judge. And all applications, including taking all the samples during these procedures, are carried out by the forensic medicine physicians. Approximately 600 forensic medicine specialists are working in the field. In this presentation, our aim is to draw attention to examinations of skeletonized bodies or skeletal remains as a part of forensic anthropology, which is an important component of forensic sciences, and forensic medicine practices within the framework of legal legislations in Turkey and indicate suggestions in the related field. Exhumation is a process of removing the dead body by opening the grave. It can be performed due to burial without required procedures, doubts about the identity, and controversial results of the first autopsy, suspicious decisions on the manner of death, allegations about false treatment and public health, and problems about indemnity. In a forensic context, exhumation typically refers to the removal of human remains, whereas the excavation refers to the removal of other buried evidence, such as drugs, weapons, or material associated with human remains, such as ballistics, ligatures, clothing, and personal belongings. However, in Turkey, there's no distinct separation between them. Human remains can be removed from the existing grave with the exhumation process, or it can be found by chance during a construction work or showing a location. On the other hand, some cases, skeletal remains can be found in very different conditions, such as in water, buried in the ground, hidden in certain areas, and dumped in a forest, for example. When we come across the skeletal remains in an exhumation process, there are several questions needed to be answered. Firstly, we need to know that if these remains belong to a human or an animal. If they are human remains, are they from an ancient burial or a recent burial? If we have a recent burial, is there a forensic background, or is this a ritual burial? If we are dealing with a forensic case, how many people located in the gravesite? Nonetheless, systematic recovery and recording of all the evidence through careful and complete exhumation of either a single, multiple, or mass gravesite is fundamental. There are several differences between trying to investigate a previously located grave and an unknown grave. Both require high level of expertise and knowledge of research techniques. At the beginning of the exhumation and excavation process, the most important findings are the findings when the grave is, or the area where the body is buried, is first opened. By examining these findings, it will be possible to determine whether the remains belong to a human, physical characteristics such as age and sex, as well as the cause and time of death, or whether the deceased buried randomly or after a certain burial process. Questions about identification, time, and cause of death are usually answered in the laboratory process. Yet we need to have general knowledge about age and sex of each remains to determine how many people we're dealing with. And also we need to distinguish these people without disturbing their position. In forensic cases, direction and position of skeletal remains are not always paid attention. However, position of the dead body can give us important information if the case is archaeological or forensic. In order to determine if the case is forensic or not, it is important to know traditions or cultural rituals or belief systems. Also, we have to keep in mind that the bodies might have been carried to another location, so this will not be the first grave site. During a forensic archaeological crime scene investigation, in the search for burial evidence, witness statements should be taken into consideration. In cases where information exists that a body or other evidence is buried, investigators will attempt to obtain statements from anyone who may have witnessed to the burial to assist them in narrowing down a search area that may contain a suspect disturbance or gravesite. However, it is not uncommon for a witness to indicate that a general area where a grave is supposed to be, but be unable to pinpoint the exact location or the burial. As in visual clues and basic principles, the laws of stratigraphy indicate that even the slightest disturbance to the ground will lead to stratigraphic changes in the soil. The fact that the stratigraphy has been altered potentially allows archaeologists and anthropologists to identify changes to the ground surface contour, soil color, and/or vegetation growth, and consequently develop a theory as to what has occurred at a particular site. As in surface changes, an assessment should be undertaken using a line or grid searches of landscape while looking for surface scatters of potential evidence such as ammunition, displaced skeletal remains, and/or parts of clothing or personal items. Remote sensing such as aerial imagery, spectral analysis, light detection and ranging, and thermal imaging, or geophysical techniques, use or search or cadaver dogs, or topsoil removal can also be used depending on the case. Crime scene investigation and exhumation require technical knowledge and technical methods, but in practice, unfortunately, we see that most of these existing techniques are not used in many cases, and rough methods are used. Of course, the effective time and cost is seen in these practices. On the other hand, such techniques help both to conduct crime scene investigations correctly and to reach more accurate results in the subsequent investigation stages. When we confirm that the presence of a grave or human remains, the analysis of skeletal remains usually follows the same principles and objectives as in the case of a fresh body. Dignified management of the bodily remains and identification of the remains, assessing the cause and manner of death, and using data methods, the time since death, and contributing these reconstructions of the circumstances surrounding the death. Recently deceased, partially skeletonized, or completely skeletonized bodies require an interdisciplinary approach. The forensic doctor responsible for the case has to work with the other specialists. In the case of skeletal remains, this requires a forensic anthropologist. The forensic anthropologist takes the lead in assessing skeletal trace for the purpose of identification of or to find and interpret signs of pathology and trauma. The forensic anthropologist may also collaborate with the forensic doctor in coming to conclusions about manner and cause of death, and using data methods to time since death. Forensic anthropologist determines manner of death by analyzing trauma, where the forensic doctor determines the cause of death. However, when it comes to the crime scene, the cause of death is not decided there anyway. A general framework is determined. What needs to be done at the crime scene is to open the grave in accordance with the rules, to evaluate burial type, to remove the person or people from the grave correctly and completely, and to send them for examination. As you can see here, an important use of right methods rather than making a reckless investigation in order to reach results quickly. When two or more body parts are found, it should not be automatically assumed that the separate body parts belong to the same person. This determination should be only made by a forensic doctor and/or forensic anthropologist. If it appears that there are multiple deceased, the exercise of recovering the human remains may follow that of a disaster victim identification procedure. DVI is an incident classified as a mass fatality incident when it involves 5 or more casualties and is subdivided into major, mass, or catastrophic, depending on the total quantity of victims. An MFI may be local, national, or more commonly these days, international, depending on the countries of origin of the victims. They may also be closed or open incidents, depending on whether the number of casualties is known or whether the identities of group involved are known. Each phase of DVI needs a multidisciplinary action. Forensic doctors, forensic anthropologists, forensic archaeologists, crime scene investigators, biologists, palynologists, veterinarian, and the list goes on, should work together every step of the way. The use of radiological imaging has been proven beneficial to the identification process in many ways. Of course, postmortem CT is a rising star. Postmortem CT is an extremely useful investigative tool in a mass fatality incident, particularly for the purpose of disaster victim identification. The rapid and accurate identification of individuals is of primary importance for both legal reasons and for relative peace of mind. Transporting bodies to clinical CT scanners following an MFI is possible, but can often create a number of logistical issues, particularly if the incident occurs in a remote area. Mobile postmortem CT scanners in a forensic setting might be a solution to these logistical issues. According to Minnesota Protocol, the recovery of human remains should preferably be under the supervision and advice of a suitably trained forensic anthropologist if the body is skeletonized, and/or forensic doctor if the body is fleshed. Knowledge of forensic archaeology is also valuable in understanding the toponymic processes at the scene. Expertise in forensic anthropology and/or archaeology may assist in the recovery of burned, fragmented, and buried remains. However, in Turkey, most of the forensic cases, crime scene investigations are often performed by police or other personnel without training or education in human anatomy. And thus, there may be challenges in identifying body parts and/or skeletal elements. As you can see in these pictures, falsely conducted crime scene investigations can lead to contamination as well as the damage to the bones and inaccurate trauma analysis. As a result, we suggest that multidisciplinary actions should be carried out from the start of the exhumation process to the laboratory analysis. Each research phase needs different kind of expertise. That's why, depending on the case, forensic doctors, forensic anthropologists and archaeologists, veterinarians, forensic geneticists, and forensic entomologists should work together. The university structure in which forensic medicine and forensic science professionals work together at the academic level is a good example in terms of interinstitutional cooperation and multidisciplinary approach model. Required experts should be invited to the scene. Methods such as postmortem CT should be used conducting an investigation, and all uneducated personnel should be trained. Thank you for listening, and thank you for your attention.
Thank you very much, Elida. It's a very comprehensive talk in a complicated issue. So we have a few minutes to discuss over this issue. So please, Professor Harrison.
But we agreed on. I guess that I can't give you the exact number, but it is so small. It's just like maybe 10%. It's just because, yeah, unfortunately, forensic medicine specialists, the only people go to crime scenes. That's why we kind of have this problem about the crime scene investigation and examination of the postmortem system. But I'm doing master's in forensic anthropology, and I wouldn't have the knowledge. I know that I don't have a lot of experience, but I wouldn't have the knowledge that I have right now without my master's education. So I believe that this would be in, as fellow colleagues of myself, this would be an education to my colleagues to, perform in the field.
Thank you very much. Is there any other questions? All right. Thank you. All right. So thank you very much, Elida. So it's time to move on. Fourth presentation is going to be online again. Is that correct, Soto? Okay. So if we are ready, we can connect to our fourth speaker, who is in Bulgaria, I believe. Hello. Hello, Karastoyanova. Sorry about misreading. It's a pleasure to have you over here. Now it's time to give your presentation.
Yes, thank you. Hello, we will present both of us. It is an interdisciplinary study. I'm the anthropologist side, and my colleague is from the osteozoology of the bones from this find we have. We're gonna present a find from Kuzareva Mogila. It is from Chalcolithic period. Sorry, yes, it is located in southeast Bulgaria. It is near Burgas nowadays. Burgas, it is a small town nowadays, town Kableshkovo. The site is located here near the Black Sea shore. It has a long history. It has layers from the Neolithic to the early medieval period. But most thicker layers are from the Chalcolithic and Neolithic period. It's 5,000 years BP. And this is the flourishment of the civilization of that part. Sorry. Yeah. Okay. The site is interesting that there is found settlement layers and the necropolis connected to the settlement layers. You see the characteristic Chalcolithic burials on the site and the specific ritual used in Chalcolithic burials. This is the plan of the necropolis. Our materials comes from this light blue area. This is a separate area in the necropolis which shows a little bit different burial ritual, which the archaeologists, the investigators of the site, Petya Georgieva, think it is section of the necropolis which were used from different population, different people from the indigenous population from the Chalcolithic period. The site is the necropolis develops in the early and late Chalcolithic period. We have some problem in dating our bones. Possibly they come from the late period of the necropolis. As you saw in the previous slides, normal Chalcolithic graves in the necropolis has many materials which provide data for dating of the burials. Our burial has no material, so for now we still date it in the big limits of the high limits of the Neolithic period. The skeleton is relatively well preserved in according to the other materials from the necropolis, but the bones are highly fragmented. Anyway, the sex of the individual identifies as a male one. You see the mandible and the sciatic notch, great sciatic notch. The age of the individual identifies as a young adult, about 18-20 up to 25 years of age. You see the restoration of the skull. You see how fragmented it is. After restoration of the skull, we found on it different markings, which are with traumatic provenance. One of them are here on the right parietal, others are on the left parietals, and the big hole on the left parietal also. Here you see a cleaned skull with the areas when we found the marks. These which are on right and left parietal near the coronal suture doesn't penetrate the brain case. The other one which is on the left near to the middle of the left parietal penetrates the brain case. Here you see the measurements of these marks before cleaning of the material. Yes, the animation doesn't work very well. Okay, you see here a close view, microscopic view from the defect which we found on the left parietal near the coronal suture on the cleaned bone material. And here you see the measurements of the big hole on the left parietal. And here this this big hole on the left parietal. All these marks are survived from the individual. You see well covering of the new bone formation on the margins of this defect. On the inner table of the skull, on the big defect, you see here we have a splinter which wasn't removed in the previous period when the individual were alive. It was survived from him, and well survived from the individual, and he lived some period after he obtained this defect, and some time this event passed. On the side we have many finds which give us idea that in this period people have some idea about the anatomy of the human bones, human skull bones. There are found several these rondelle which were cut from the skull. Other finds are from cutting of the bones before burial. We have also these real trepanations which were performed those times. There were such trepanations, but the person we investigated, even he had a splinter on the inner table of the skull. He wasn't trepanned, and he survived without clear surgical help on this skull. Now I am giving a word to my colleague who will clear the event.
Yes, I will start with methodology. We suggest that these traces are made by punctures from a large carnivore, and for this reconstruction of the attack from the carnivore, for the pit marks, they were compared with bites of 2 skulls selected from the collection of the National Museum of Natural History in Bulgarian Academy of Science. From left side, you see a skull from a large male bear killed in 1996 by a hunter. And there is a reward from a hunter competition in Berlin for the big size of the animal. And from the right side, there is a second skull from a male lion, died in 1955 in the Sofia Zoo. Still very large animal. First, for the methodology, we needed to clean the deposits in the pit marks, because as you can see in the picture before, you can't see actually any pit marks because of the deposits inside may be made by some organic stuff. For this reason, we use an ultrasonic cleaning device in laboratory, which were made by a professional restorer, Gergana Baykusheva. With this device, she just removed the deposits without touching the surface of the skull. And the result is that we have very clean traces that we can examine. I use molding for the pit marks, because these moldings can give us a more like a 3D shape of the pit marks. And in that case, we can compare it with the teeth, with the punctures of the teeth from the animals. Why we think that this is from large animal carnivore? Because of the scale and measurements that we took and compared with these measurements are compared with large carnivores. Why we think that that can be from these large carnivores? Because not only because of the size of the pit marks, we think that it's something larger than a dog or a wolf. And we think that probably we suggest that it's from a lion or from bear, because these animals exist in this period in Bulgaria. In an Neolithic period, we have some remains from lions, and during the favorable climatic phase corresponding to the Neolithic, the lion had maximum expansions in the Balkans in this time. In Europe, it disappears around the 1st century AD, according to some pictures. Sorry. In Bulgaria, we found from this period 3 sites that we have some remains from lion. The first one is Duranculak, which is in northeast Bulgaria. The second one is Guleamo Delchovo, which is in the Balkan Mountains, but still pretty close to the Black Sea coast. And in this Chalcolithic site, there is not only a remains, but this zoomorphic statue that you see on the picture. This zoomorphic statue shows that probably they have some connections with the lions in this time. And the second site is south from Kozareva Mogila site. This is a Sozopol site. There are several remains from lion from these periods again in actually very close to Kozareva Mogila. And the brown bear, the European brown bear is widespread everywhere in Europe and in Bulgaria. There are remains from Neolithic till Middle Ages in most of the archaeological sites. We made some kind of reconstruction of attacking of the bears and lions. According to studies, there is a study which assumed summarized the cases of recent attacks of bears and lions, and the bear attack pattern is one of the best studied in forensic medicine. Biting the victim also common, and a bear attack and different degrees of minor and major injuries, predominantly located in the upper half of the body, especially in the head and the face. And there are a lot of cases which the main damage is mostly on the head and the upper limbs. Bear tend to attack the victim's head, causing wounds and fractures in that zone mostly. And the lions, their attack pattern, it's almost the same. But this is usually usually they attack the cervical vertebrae, but also there can be damages on the head, some punctures, perforations, or cortical fractures caused by several bites may occur. In our reconstruction, we use these skulls just to fit to the pit marks. And in this attack, we suggest that the position of the pit marks suggests that the person was sitting, squatting, or laying down when this happened, or was knocked down by the animal. Why we think like that? Because always when we try to fit the pit marks to the skull of the animals comes like over the head, over the skull of the human. And in front, that's why we think that probably the human was sitting or laying on the ground. The animal bit the victim at least 2 times, only on the head, because there are no traces of postcranial bones. We think that there are 2 more bites than 1 only, because the pit marks doesn't fit only to 1 bite. And the human survived from this attack. We use modes again just to show from which animal probably are the pit marks. And in this case, from your left side, you see one of the pit marks, one of the big pit marks, and from right side is the mode. This is a print from the upper molar tooth of lion, and you can see that the size and the shape, it's not the same, but almost the same. That's why we suggest that probably this pit mark is from this tooth. And of course, we have a lot of unsolved problems about our suggestions, our constructions. The first thing is that man was younger when he was attacked. That means that the measurements may not be accurate because the skull is smaller. The reconstruction suggests that it was more than one bite, only because the bite doesn't match completely. That's our problem. But this problem could be due to the incomplete skull, the smaller size of the skull when he was attacked, and healed wound, which changed the traces. The traces are, they're a little bit fused. And there are no traces of attack in the postcranial bones, but that could be because the wounds may not affect the bones, only the flesh.
Thank you.
And thank you for your attention.
Thank you very much. Thank you. Very nice presentation and meticulous work on this skull. So we have a few minutes to discuss over your case. Any questions from the hall, please? Okay. Thank you very much. See you soon.
Please. Thank you.
So now we're going to move on to the last presentation. It's going to be video presentation. So if we are ready, we can connect to the video for the last presentation.
Paper by me and some colleagues whose names I'm going to refer to very soon. The team, first of all, of our project, or part of the team, starting from Dr. Nikolaos Montozoglu, the head of the CT scanner and the Medical Center of Athens, whose general financial director is Mr. Emmanuel Markopoulos, and the director for public relations, Mr. Kostis Hadjigiannakis. I am the coordinator of the project, and then we have the former curator of the Egyptian collection, Miss Helen Tourna, archaeologist, Professor Emeritus Stefanos Geroulanos, historian of medicine and also professor of surgery at the University of Zurich, Mr. Panagiotis Lazaris, conservator of the collection, and Mr. Konstantinos Kouvaris, medical doctor, forensic pathologist, and anthropologist, who is working for the Red Cross. So the aim of our project is the Egyptological, medical, and forensic study of 10 Ptolemaic mummies from Panopolis, from the modern area of Egypt called Akhmim. Here is a part of the scientific team with Dr. Nikolaos Montozoglu and his team of doctors, Dr. Ioannis Pandazis, Mr. Ioannis Pandazis, a medical doctor, specialist in mapping the mouth cavity and teeth. Mr. Kyriakos Kalamboukas, also medical doctor, and Mr. Michailidis, also medical doctor. Here is our coroner, and Mr. Lazaris and Mr. Na, etc. Two more members of the team of doctors of Dr. Nikolaos Montozoglu are Mrs. Stavroula Kyriazi, medical doctor, and also Mrs. Kleanthi Kalogerakou. Talking now about the mummification ritual, and starting with the virtual reality picture. Here you can see actors playing the roles of the priests during the final rites for mummification. Our paper is written by me, Dr. Nikolaos Montozoglu, Mr. Konstantinos Kouvaris, and Professor Emeritus Dr. Stefanos Geroulanos. You can see, of course, the abstract in the abstract book, and the key words are Egyptian mummies, Ptolemaic period, Panopolis, embalming techniques, mummification, excerebration, diseases, death, putrefaction, prosthesis, computed tomography, CT, paleoradiology, paleopathology, and forensics. We know about the chemistry of mummification, that they were using special substances, especially natron, that was a combination of three extremely hygroscopic substances, mostly of sodium carbonate and sodium carbonate with hydrogen, and with traces of sodium chloride and sodium sulfate. That was removing the water from the tissues, as well as other substances that were found in the resins used for mummification. And all of these substances have intense antimicrobial and antibacterial properties. For the chemistry of death and the putrefaction, also we know a lot of things, and I don't have time to refer to all this. The Mummy Project of Athens, I'm going to present you briefly our objectives, scopes, and planning, giving also simultaneously a short history of the project that is taking place between the Hellenic Institute of Egyptology, the Athens Medical Center, and of course the National Archaeological Museum of Athens and the Egyptian collection, which is hosting the mummies. Also, we have the International Hippocratic Foundation. For the moment, we don't collaborate with the Athens Forensic Authority, and we collaborate also with some transporting and insurance companies like Orfeve Inoglu, a special company for transporting antiquities from one country to the other. Our objective, scopes, and planning, we are going to say a few things about this. And of course, we always respect the dead, and we scan the mummies in a humanistic way. We have Egyptological objectives, archaeoanthropological objectives, medical and forensic objectives, and educational and cultural that I'm going to analyze in the next few minutes. The first scope is the full Egyptological study of the sarcophagi, their inscriptions, the funerary treatment of dead bodies, and of their clothes. The comparison with other published or unpublished sarcophagi and mummies. The chemical and physical situation of the wood of sarcophagi, clothes, and substances found from a conservator's point of view. The full publication of the finds and conclusions in a splendid volume that we are planning to publish in the future. Also, from the medical point of view or the forensic point of view, determination of the age at death and the causes or cause of death, if possible, of course, various ailments, syndromes, etc. Study of the skeleton, the remaining muscular mass and the remaining organs. Study of the teeth and odontograms. Study of the wear on various bones, teeth, and the flesh in order to conclude about their health condition, diet, environment, etc. Reconstruction of some of their faces using special software and forensic anthropological techniques. We are planning also a possible exhibition at the premises of the museum, like the one that was very successful in the Zagreb Museum before about 10 years in Croatia. A series of lectures and guided tours for the general and some specialized public, the publication of the purely medical and forensic material by Egyptologists and the team of Dr. Nicolaou Bondozoglu in specialized radiological and medical journals. We have published already 5 papers, and we plan to continue with some more mummies, inshallah, God willing. Let me show you our journal, the journal of the Hellenic Institute of Egyptology, that we are planning to enter Scopus very soon. This is the 4th volume, and it was dedicated to the Athens Mummy Project. And the last paper was published in this. You can find the rest of the papers if you visit my academia page, Academia İYYU, Alicia Maravella. Computed tomography scanning. This is the scanner of the Medical Center of Athens. Siemens Somatom scanner. It's a non-invasive study. Actually, our permission from the Ministry of Culture is only for non-invasive studies, so we don't have the permission to get DNA samples. And this shows us the preservation of the mummy. This is the mummy of the priestess, on which I'm going to focus mainly today. The age at death and the cadaveric height, sex confirmation. Probably we can find some interesting finds that show the sex, so that we can confirm the inscriptions on the coffins, possible causes of death, not always, the way of mummification, ailments, syndromes, etc., teeth conditions, and we use smart informatics and reconstruction with panoramic 3D mapping. So we have an interdisciplinary field and interaction between smart informatics, paleomedicine, and Egyptology. Here you see the mummy of Sekhem, another interesting mummy of our sample. Siemens Sensation 64, the somatom, is a CT scanner of 64 sections. You can take many slices and take details. Slice thickness 0.6 millimeters. We get 5,600 slices per scan and use 10.7 gigabytes per scan. And some more technical information. The mummies of this project were examined using a multi-detector CT scanner using special software that you can see here. I don't have time to stay on all of this. 3 scans were performed for each mummy. The first one was a whole body scan from head to toes, acquired at 120 kilovolts and 250 milliampere per second, times second, with a slice thickness of 0.6 millimeter. And you can see the rest of the details, together with more technical details. The multiplanar reconstruction involved the process of converting the data acquired from axial CT images to coronal, sagittal, or oblique 2-dimensional images. And we usually show the MIP images, and we have, as you will see, excellent results. Here you see the mummy of Tutankhamun, the body of Tutankhamun, and the reconstruction of his face using special forensic software. Using for this silicone. Here you see the mummy of King Seti I and that of King Thutmose IV in the Cairo Museum. Now I'm going to introduce you to the 10 mummies of our sample, very briefly. I will say some details about some of them, and I will focus my attention in the priestess that was the actual dilemma of this project. Here you see the mummy number 3340 of Hapi or Hor, the only one exhibited in the Egyptian collection of the museum, early to mid Ptolemaic. All of these pieces come from the Dimitriou collection. Ioannis Dimitriou was a Hellenic expatriate who was living in Cairo and Alexandria, and he was rich. He obtained a very good collection and offered it to the Archaeological Museum. The coffins with their mummies were found in excavations by Dr. Gaston Maspereau in 1884 at the necropolis of Chemis, and were officially purchased by the wealthy patriot Ioannis Dimitriou, who offered them to the newly inaugurated National Archaeological Museum. The second mummy has also a very interesting coffin. It belongs to a woman, car-ready busted, early to mid Ptolemaic. It has not yet been scanned. It has inscriptions offering formulas. The third mummy, 3342, it belongs to Thao, the son of İYYU and Lady İYYU, early to mid-Ptolemaic period, and this presented us with the first surprise, because as you can see here, from outside you can see it looks like a mummy, but the inside is completely different. Actually, there is no cutaneous part or musculature. There are only bones, and we found 3 different femoral bones that were broken or cut. So inside the mummy, inside the wrappings of the so-called confected mummy or pseudo-mummy, although it's ancient, everything is ancient here, you see a very good technique of a lot of linen wrappings with resin coatings, thick resin coatings between them, that shows something, creates something that looks like a mummy, but unfortunately there is no mummy inside, only bones. And we have 2 different ways to hold the whole confected body together. We have canes made of reed. You can see here that it's reed, and also canes or sticks made of palm tree. 3 femoral bones we have, as you have seen, and pieces of wood. One made of reed and the others made of palm tree. How we have defined this? The reed is very easily to be seen. It has these knots, and the palm tree, we got the scanning of the cross section, and we scanned also a modern contemporaneous branch of a palm tree. So you can see it's actually palm tree. And this is a technique that was used in antiquity. You can see also the pattern that is so-called H-shaped pattern of the outer linen bandages with heavy coatings of resin, and you can see what is visible inside. This paper is published by Kalamboukas, Kyriasi, Maravella, et al. You can see it in my academia page. The fourth mummy belongs to Sekhem, son of Takhor, mid-Ptolemaic to Roman period. This was also an interesting mummy, in which we found one of the most interesting finds was the placement of his internal organs in a canopic package inside the mummy, and this contains actually the 4 main or holy organs that were preserved inside the body, or mostly outside the body, in 4 special jars called canopic vases or canopic jars, each one under the protection of a son of Horus, of a special divinity. And you see here the different organs. Here you can see the stomach with something inside, either an amulet or a piece of bone, the liver, the intestine, and the lungs together with different pseudo colors to show them. The mummy of Sekhem, in which we found also a small object of very high density, optical density, about 2000 Hansfield units at the area of the neck, either a periapt, an amulet, or some bone. And we found another very interesting find in this mummy, the second in the international bibliography. We found a tooth packing that shows that ancient Egyptians were using dental praxis. So there is a dental pack between the right mandibular, second premolar, and first molar, number 45 and 46 in the language of dentists. So now, this is also published by Pantasis, Pruna, Maravelli et al. Let's go now to number 5, 3344. This belonged to a child, most probably male, a small boy, whose name was Tekha or Tekti or Tekhor, as we can read from the inscription, where some of the hieroglyphic signs are not easily discernible. You see it has smaller dimensions, and the finds here were not so impressive. However, they were interesting since we detected missing toes, something that happened post-mortem, dental disease only of molars, and we also found that it was subjected to a transperineal evisceration. That is, that they removed the internal organs through the perineum, not through a cut at the left hypochondrial side of the body. You can see the wear on the outer bandages. Some part of the cranium are visible, and as we unwrap the mummy, as we continue to proceed inside, you can see the toes, and you can see that some of the phalanxes are missing. An interesting find is that the age was determined by considering that there was an arc without teeth. The front teeth had not yet erupted. That is characteristic of about 8 years of age. Another mummy belonging to a child that has not passed yet through the scanner. We have scanned 5 of the 10 mummies. 5 more remain to be scanned. However, one of them will not be scanned, as we see, because as we shall see, because it's in very fragmentary situation. 3346 was also a very interesting case belonging to Tati Thedamun, son of Onufris and Lady Sonek, and with a very interesting inscription. I give you to see what it says. It asks the gods to offer food and other offerings to the spirit of the deceased. Tati Thedamun, the excerebration has not taken place. We found out that the ethmoid and the cribriform plates were intact, brain intact, shrunken at the occipital bottom of the skull. We found dislocation of pelvic bones post-mortem, dental disease, heart inbound in the body, and also a most interesting syndrome for the first time in the bibliography, so-called silent sinus syndrome or chronic maxillary atelectasis. And this could have resulted in the deformation of the face of this individual because his eye would have fallen down and he would look ugly. We found also remnants of a Fayence bed shroud, as you can see both on the head and on the body. And we used virtual reality, augmented reality techniques to reconstruct the Fayence shroud. It should have looked something like this. This is a Fayence shroud of the Egyptian collection of the National Archaeological Museum in Athens that is almost intact. So the body was covered with this. We go now to really the one that will not pass from the scanning. Another lady of the sample. There are 3 ladies only. The one will not pass because it's in very fragmentary condition. And now let me go to the last part of my talk with the ritual dancer of Min, the lady Takheret Menu, daughter of the 4th prophet of Min, Iret Koreru and lady Aset Nekhebut. This is a most interesting case because although we have been expecting a very sophisticated embalming method, unfortunately, we have found a very strange situation because what we found inside was that there were not so many muscular and cutaneous parts, but mostly bones, not very anarchically placed, and as you see, no third femoral bone. However, the situation was strange. First of all, there was an attempted excerebration that was failed, and we find remnants of the brain shrunken at the occipital bottom of the skull. We found disordered and fractured bones that happened post-mortem. The corpse was found well after the putrefaction started, and we found other interesting finds that I'm going to analyze you very soon. Why it was given to the embalmers the body after putrefaction started? Probably the most proper reason for this is what Herodotus, the father of history, writes, that concerning the women of the most important men, they don't give to the embalmers immediately, but until 3 or 4 days pass, because they were afraid of necrophilia. Perhaps the facial reconstruction of this mummy could provide a hint of this reason, depending on her beauty. We are going to do this in the future, so we are going to be sure probably she was also beautiful. Actually, we found prosthetic surgery, like plastic prosthetic surgery, because we found in the eye orbits masses of linen and resin, as well as in the nose. Eyes and nose are from part that start to putrefact after death. According to two scholars, during the Ptolemaic and Roman periods, the poor quality of mummification and the fact that the bodies were in an advanced state of decomposition before they were embalmed often resulted in many of the bones being dislocated. Also, we found about 40 centimeters of palm wood that was supporting the cranium and was passing through the foramen magnum, almost parallel to the vertebral column. Here you can see the remnants of the brain. And here you can see a cross-section of the scanning of the palm wood. You see typical cross-section of palm wood, surrounded by linen soaked with resin. And here you can see more photographs with the pseudo colors to see the palm wood, the remnants of the brain, and also you can see these parts here, these parts that are resin, and this is the prosthesis I told you. They try to reconstruct the nose, so they place resin and linen. And here you see also the wrappings that are not so many. Although there is a lot of resin. Using sticks like this and also X-shaped props like this is common in all periods. This comes from the third intermediate period, if I'm not mistaken, and from Del Medina, from a burial of Del Medina. This is an abdominal prop, so they placed in the abdomen to support the empty body, as they were using also to put inside linen, sawdust, etc. Here I show you some of the hooks, metallic hooks that were used to make the transnasal excerebration. They were breaking the ethmoid and went into the brain to steer it and put it out. And we go now to the mummy of the Royal Ontario Museum, belonging to a baby of probably between 7 and 11 months old, whose head is also supported by a palm wood, a smaller palm wood, as you can see here. If we start to, you see the displacement and the disorder structure of the bones, especially in the abdominal and the stomachic area, and the wood also here. And if we go to unwrap the mummy virtually, we can see the details. It's the only mummy that found in for whom we found the hands in the Osirian position, probably because she was a priestess. And here you can see the mummified body with the head displaced, and this that protrudes is not actually the nose, but it's the prosthetic nose. Here you can see the cross section of the wood, the piece of wood they used. The last mummy belongs to Jed Hor or Neshor and has not passed yet through CT scanning. I would like to thank you for your patience, for your invitation, and I would like to thank very much the Medical Center of Athens, the Director General, Mr. Apostolopoulos, the Financial Director, Mr. Markopoulos, Dr. Nikolaos Bondozou and his team, as well as our colleagues from the museum, the new conservator, Mrs. Argyro Grigoraki, MA, and I show you some snapshots from the scanning project and the special boxes that were used to transport the mummies. Thank you very much. Çok teşekkür ederiz. And merhaba again.
Thank you, colleagues from Greece. So we have come to the end of session 3. We're going to have a coffee break till 4 o'clock. So let's meet at 4 for the last session.
Okay. We can begin. Good afternoon. I would like to welcome you in this afternoon session, History of Medicine. And I introduced, I am the Professor Konstantinos Protoukis. I am a radiologist and Professor of the History of Medicine. And I would like to invite to speak my dear colleague Kostas Tsiamis, who is Professor of History of Medicine and Public Health in the University of Thessaloniki in Greece. So, Dr. Tsiamis, welcome.
Good evening from Greece. Thank you, Mr. President. My congratulations to the organizing committee for your work and your efforts for the success of the 4th International Conference in Istanbul. I am Kostas Tsiamis. I am Assistant Professor of History of Public Health and Migrant Medicine, but I am cytopathologist. So I am familiar with the molecular methods. And actually this presentation, this brief presentation is an alternative point of view as a result of many conversations with good friends and colleagues around the world from different scientific fields. So, molecular methods, the future of the medical world. But we have some problems. The target of this presentation, in the frame of the spirit of the 4th International Congress, is to remind to all of us the value of the collaboration between different scientific fields. Until now, we knew that without the help by the historians, we cannot approach the sources. We cannot, for example, to make a grouping of the epidemic outbreaks, because they gave us the chronologies. They are experts. We had collaboration and we have collaboration, we have networks. But the last decade, we said we don't need, not all of us, but the tendency. We said we don't need the historical sources. Why? Because we have skeletal remains and we can introduce molecular methods and DNA analysis. Do you believe that is useful for the historians if I start to explain why Yersinia pestis was derived from Yersinia tuberculosis and both of them from the Yersinia enterocolitica? I don't know. Or to explain the evolution of the microbes and the plasmids and the horizontal gene transfer and the BIOPS system, secretion system. It's very important for the evolution of the microbes. I don't know how useful it is. Do you want something really impressive? Look this. The multitoph, mass spectrometry. You can find amazing scientific reports, the Medline, with the use of multitoph in paleomicrobiology. But I will repeat it. Do you believe that this is useful for the historians? And because this session is about the history of medicine, for this reason I'm talking about the historians. So the first impression is that we are looking the same river from different banks. And we need a bridge and a common language. We use data from several disciplines, such as epidemiology, microbiology, paleomicrobiology, history, historical demography, historical geography, archaeology, and bioarcheology. A common point of friction when examining a proposed diagnosis for the old pandemics and epidemics, the outbreaks, often stems from the argument that no disease of the past can be safely defined, for example, as a plague. This is the main topic, the plague. Since it is not the same disease from the same microbe that we know of today, considering the rate and the number of microbial mutations. Yes, the microbe is not the same. For example, Yersinia pestis, for Yersinia pestis, we have amazing number of mutations the last 2000 years. But after 2000 years, Yersinia pestis remains Yersinia pestis. If we have dramatic mutations, then we will have the evolution step and we will have another species. Microorganisms evolve, but the same is not true for the defense mechanism of Homo sapiens. Although the human genome was undergone numerous mutations over the course of millennia, these mutations have not been able to dramatically alter the way in which the body responds to external bacterial stimuli. Before the discovery of antibiotics, humans of the 6th century became infected with Yersinia pestis and died by Yersinia pestis in exactly the same way like the untreated patients of plague in Madagascar before 4 years. Mutated or not, microbes have been causing the same sequence of reactions of our organism with the activation of the same organs, the bone marrow, the blood, and the same cellular elements, the lymphocytes, mononuclears, and cells, and so on. This period during the pandemic of SARS-CoV-2, suddenly we saw a great interest on the history of epidemics. Modern scientific collaborations lead to interesting and useful conclusions which are easily disseminated. We have the internet and huge databases. For example, with this keyword, plague, without filters in Medline, without filters, for example, bubonic plague, plague antibiotics, plague genomics, plague vaccines, and so on. Just this keyword, plague, more than 12,000 results. And as you can see, this is very interesting and very nice work, History of the Plague, an ancient pandemic for the age of COVID-19. So, let's accept that the fact that a researcher has access to all this data. What kind of information is needed and by whom? And how easy is it for a researcher to understand the findings of another scientific field that is different from his own? Studying the epidemics of the past possesses a challenge, and regardless of how theoretical our approach is, such studies present certain scenarios which are more or less likely to be closer to the facts and the real event. Research is multidimensional, as it draws out the data from various sources. And as a biological phenomenon, epidemics are multifactorial systems. And the typical example is the example of plague. So let's see some data on the plague epidemics according to microbiology, historical epidemiology, with valuable help by the historians. What do we know until now? We need to clarify that Yersinia pestis has been divided into biotypes based on the fermentation abilities of the bacterium. So we have these biotypes, Antiqua, Medievalis, Orientalis, and Pestoides microtus. The molecular studies allow us to further classify the strains. The usual classification is based on the analysis of the nucleotide polymorphism, the SNP, grouping modern isolates not in biotypes, but in branches. And we have 5 branches. Branches entail sub-branches and clusters, which are, and this is very interesting, which are geographically defined. The non-human pathogens of bioarcheological spread all over Central Asia, China, and Mongolia. Of course, molecular studies progress and new genomes are analyzed of the bacterium leads to the constant inclusion of new data into classifications. For example, from Mesozoic foci in Russia and Tibet. Other branches divided into the Caucasian, Caspian, and Central Asian Chinese branch. Notably, the existence of Yersinia pestis strain 91001-001, which was isolated from Inner Mongolia, is human avirulent but lethal to rodents. More recent studies examining numerous strains reveal the constant presence of the bacterium in Asia prior to the first pandemic, the well-known Justinian plague. The study of more than 500 samples from skeletal remains from Russia, Croatia, Estonia, Germany, Hungary, Latvia, and Lithuania from the late Stone Age and the early Bronze Age revealed the presence of a microbe containing parts of the sequence of Yersinia pestis. This finding leaves open the possibility of the introduction of the microbe into Europe during the migration of populations 5,000 years before Christ. We still do not know how the disease was transmitted, but it has been argued that the bacterium reached the European continent sometimes between 3,000 or 5,000 years. In fact, it is estimated that after its initial introduction into Europe, the microbe could have gradually returned to Central Eurasia during the Bronze Age. A similar study of 100 samples from skeletal remains around Asia and Europe from the same period, we have the same result. And it's very interesting, the absence of crucial virulence genes in these findings. And the first estimate is that these strains have no direct relation to the one that affected the Byzantine Empire during the 6th century AD. The studies concerning the strain that caused the first pandemic are of particular interest. The comparative analysis of more than 200 modern genomes and the genomes of the 2 pandemics highlighted the relationship between branches and different clusters of strains in the Xinjiang region of northwestern China. The same result was in the study of skeletal remains in Kyrgyzstan. Exactly the similar result. It was determined that the strains belong phylogenetically to the same branch, let's say the Asian branch. Furthermore, the basis of all strains of the first pandemic seems to have originated on the Tian Shan mountain in Central Asia. On the other hand, it appears that the strain that reached Europe began its own evolution, even though the core of its genome remained unaltered. Apparently, this is where the role of the microevolution of European strains came into play. A typical example is a very interesting study between strains from Norway, the strain of Oslo, England, the strain of London, of Saint-Laurent of southern France, and the strain of Barcelona. And they have 100%, they are identical and corroborated historical sources in relation to the spread of the epidemic in Spain, France, England, and Norway during the period of Black Death. The Eastern Mediterranean comes across as the gateway of a single entry of the disease, or even as the place where the disease was constantly regenerated. After the establishment of the disease, indeed, the Mediterranean experienced the reintroduction of strains from Western Europe and Central Europe, making the ideal place as a region, the Mediterranean region, was the ideal place for this, let's say, molecular recycling and microevolution of the microbe. Besides the impressive findings of molecular studies, however, we should not forget the main principles of classical epidemiology: the role of human activities, the population of movements, the wars, and according to our knowledge, the herd immunity. And the information of this role is not derived by the molecular methods, but from the historical sources. Even in the absence of other risk factors, trade routes and the population movements are sufficient factors for the reestablishment of disease in the region. Unfortunately, the lack of skeletal remains in the Mediterranean region, Eastern Mediterranean, dating from the era of the Justinian plagues or Black Death, does not allow us to make comparisons with the respective strains, the known strains of Western and Central Europe. Although, okay, this is a theory, it would come as no surprise if such a study concluded that the territories of the Byzantine and Ottoman Empire were affected by multiple strains of Yersinia pestis, with origin from Western or Central Europe. I think that it is easy for anyone to understand the difficulties stemming from the approach to the same issue by two distinct sciences from a different starting point. It is understood that when we examine this issue, for example, the old epidemic outbreaks, over the course of centuries, its solution becomes more difficult. It's completely different to work with a Justinian plague or the Black Death, and completely different if you work with bills of mortality and the modern plagues in England, for example. So the solution becomes more difficult. And above all, don't forget that the study of epidemics beyond the molecular methods moves along two elements: space and time. An elementary code of communication will be helpful between scientific disciplines, allowing non-specialists to understand the way scientists with medical knowledge obtain data to build a theory on past epidemics. The common code would help scholars to understand and evaluate the points related to infectious diseases in historical sources. By learning about the evolution of microbes, in our case, Yersinia pestis, historians could better understand the possible relationship between this evolution and the historical period and facts. For example, can you imagine a historical source with a very important information, for example, deaths of animals in Byzantine Empire or Ottoman Empire or British Empire, I don't care. Do you remember the description of Cantacuzenius, Emperor Cantacuzenius, in his history on the second pandemic, the Black Death, about the falling rats from the roofs of the houses? This is not a trick by Cantacuzenius in order to impress the reader of his time. This is a real fact, the famous phenomenon of rat fall. We know about this in every epidemic of plague, and it's a fact beyond the molecular methods. It is clear that the purpose of medical research is different from the historical studies. Indeed, medical research on past epidemics has been carried out from the perspective of molecular medicine, a field that can hardly be understood without medical knowledge. A well-dated, and I will finish with these thoughts, a well-dated historical source concerning an epidemic that affected the city, the potential archaeological findings of skeletal remains from the same period and the same city, and a potential finding among these remains related to a pathogen using molecular methods do not necessarily translate into de facto proof that this microbe mentioned in the molecular findings is responsible for this epidemic outbreak. Unfortunately, the more distant the past, the less likely is to reach definitive conclusions on evidence. This scenario, however, shows that the study of past epidemics, regardless of its starting point, can serve as a meeting point for distinct theories and scientific fields, bridging their differences in a common cooperation. So we use molecular methods, we don't forget historical studies, and we construct a common language because they cannot understand our findings. Thank you very much.
Thank you, Professor Tsiamis. And we have the first question, Professor Kalpul.
Can you hear me, Professor Tsiamis?
Do you hear us now?
Can you hear me now, Professor?
Yes, it's better. Thank you.
Okay. I thank you very much for this very, very interesting important talk. Maybe my question is not the correct question.
Sorry, I can't hear you.
I think the microphone is on.
Is that okay now?
Yes, now it's okay.
Perfect. Maybe the question that I will ask is not directly related to your expertise or the area, but it came to my mind after your perfect presentation talking about the lineages of and the DNA changes in the microorganisms. Do we have any indication of some microorganisms that ended up, that we do not have them now? Are there any microorganisms that their DNA is absent now, but we see them and find them in the ancient times? This was the question. I don't know whether you have the right answer or somebody has the right answer. Thank you.
Thank you very much. It's a very interesting question. As I know from my research and my reading, we have not data that we have not this microbe today. According now, okay, let's say that we have not the virus of smallpox, but this is another issue. But according to your way of thinking, and this is very interesting, we cannot say 100% that this microbe is disappeared.
Okay.
Actually, sorry, we have, let's say, updated microbes. We have the same strains, or not the same strains. We have the same biotypes, and we have small differences in the genome. But as I said, Yersinia pestis is Yersinia pestis. I don't care if this is Yersinia pestis Kim or another type. If we have dramatic mutations, we have another species. Like from Yersinia pseudotuberculosis, after dramatic mutations, we have the born of Yersinia pestis.
Okay. Thank you. Any other question, Professor Tsiamis? So I would like to thank you, Kostas.
Thank you very much.
I know that what happened. Okay, and I know that Dr. Tsiamis has another teleconference, so we go to the second speaker, I invite Dr. Marios Papadakis.
I cannot hear you.
Now?
I cannot hear you. Can you hear me?
Now, do you hear me?
No.
Πού δείχνει εδώ? Όχι, δείχνει στην οθόνη ότι δεν υπάρχει μικρόφωνο. Okay. So, Dr. Papadakis is is a plastic surgeon and historian of medicine. We collaborate many years. And,
I cannot hear you. Can you hear me, Mr. President?
Yes, I can. Okay.
So should I go on? Okay, yes. So thank you very much. Many greetings from Germany. I'm Marios Papadakis. I'm a general surgeon and a plastic surgeon. I would like to thank the president for the introduction, although I cannot hear him. My topic today is about the first description of mesenteric ischemia in the 2nd century AD. Mesenteric ischemia is an ischemic and inflammatory injury caused by inadequate blood supply of the gastrointestinal tract. There are two clinical entities of two causes, mesenteric artery ischemia and mesenteric vein occlusion. I bet I'm the only surgeon in panel, so I have to say something about the disease. It's not an easy condition. It's a life-threatening emergency with a very high mortality rate, reaching up to 60%, due to occlusion, congestions of bowel ischemia, infarction, and fatal necrosis. And this part of the bowel has to be removed. There are many papers about the history, the evolution of mesenteric ischemia. That's the most famous one from Boley, published in 1997 in the Surgical Clinics of North America Journal. And here we can find several milestones about the evolution of the disease. So according to this this review, the oldest reference to mesenteric ischemia dates back to the 15th century and is credited to Antonio Bernviene from Florence. The reference is included in this book titled A System of Medicine, edited by Albut and published in 1896. There are several other milestones, but we go direct from the 15th century to the 19th century. Hodgson, 1815, Tidman, 1843, Virchow, 1847 and 54. And the most detailed description is included in this book here. It's written in German. It's from Lytton, 1875. But the most important milestone was 1895, when and treatise from Eliot has been published. He was the first one to summarize his experience with 20 cases. He was the first one to list symptoms and provide numbers and frequencies. So we see here this table from 20 cases. He states as the most important symptoms pain, most common symptoms pain and vomiting and bloody stools. But he was also the first to describe the surgical treatment from the two cases. One case passed away, but the other survived. It was a 25-year-old patient with gangrenous intestine. He was treated with artificial anus and reanastomosis after 2 weeks, and he developed a fistula, but he survived the whole thing. So that's the first description of the causes of the disease, the symptoms, and the therapy. So, but except for these 6 milestones, there is a gap in the literature. So we don't have anything before the 15th century. But the thorough review of the ancient Greek literature reveals that Galen in the 2nd century was already aware of this disease. Galen is famous. He is the most prolific ancient physician, and his work was highly influential during the Middle Ages. His works are referred to as Galenic corpus and consist of over 120 treatises. And we are lucky that Carl Kuhn compiled this in 22 volumes and published them in Leipzig between 1821 and 1833. But the problem is that all books are bilingual, so Latin and Greek. So they have not all been translated in English. So in the 8th volume, chapter number 6, there's a treatise titled On the Affected Parts, and there we can find a thorough description of the obstruction of the bladder, of the urinary bladder. But followingly, Galen refers to a similar condition that affects the intestine and the stomach. He emphasizes that this should always be taken into account, as it's very serious, and she was aware of the systemic symptoms and the unconsciousness causing to the patient. So here is a description. That is from Kuhn's book. That's in Greek. I have translated here in English for you. So Galen said, I ask you to remember it forever. In the so-called thrombosis, not only those that occur in the bladder, but even more those that occur in the intestine, stomach, and chest, it happens that the patient becomes pale and loses his consciousness and has short, barely palpable, and frequent heartbeats, loses control of both the mind and body. The same happens also in many cases of major muscle injuries. Many wonder what the etiology of this event is and whether our own blood can also alone cause so much damage when found outside the blood vessels, followed by sepsis and soft tissue necrosis. So from these descriptions, we understand that Galen was fully aware of the systemic inflammation response induced by mesenteric ischemia and its severity. He had sufficient clinical exposure to the condition. And interestingly, he was not alone. He was not the only that observed this disease. Thrombosis and ischemia were also observed by others who appear to be debating over the causes of bleeding and inadequate blood supply. So to conclude, Galen was aware of mesenteric ischemia and its severity, always in the 2nd century. So there's a gap between the 2nd and the 19th century, and that's why we need further research. So treatises and oral histories should be examined to reveal more information about the perception of mesenteric ischemia during this period between the 2nd and the 19th century. Thank you for your attention.
Thank you very much. It's very important for the history of medicine to understand and to find the first point of every surgical or every medical practice, because for many of medical practice, we believe that began in the 19th or the 20th century, but we find that in Byzantine medicine or in ancient Greek ancient medicine. So, any question about the work of Dr. Papadakis? Thank you very much, Marius. And now I would like to call Professor Kalfoglou as he come to tell us about toxicology in antiquity with collaboration of Sotirios Kalfoglou and Ali Sefik Kyoprolou.
Now, are you ready to ask me all kinds of questions? So, at that time of the day, where you're all very tired, I don't want to bore you more. I will be short and try to be not very boring anyway. Well, it was not easy. Well, actually, I'm teaching toxicology. I'm teaching forensic toxicology. it was not very easy to start writing something about the history of toxicology. This was very new for me, and it was very interesting. We know, and we all know, everybody knows, that always people hunted using poison. We're talking about 30,000 years before present, where we know that people were using weapons, stone weapons or metal weapons, where they had poison to be able to kill the animals easily. Okay, let's go to the second slide. Oh, okay, I'm sorry, okay. Excuse me for the eyes. It is formatting the slides. The eyes are with a dot. I'm sorry for that. Well, from Babylonians to Greeks and Romans, poison was used, but and actually was used because of that weaponing, as I told you before. And actually, the word toxicology comes from toxon, which is the real weapon name for the arrows. But is this the reality? Where do we know? Do we know? Or do we imagine? We don't know. Do you know? Nobody knew up until these people decided to work on this. And this is the biomedical sciences in archaeology part of this topic. The first lady that you see on the left is an anthropologist, archaeologist. And she asked the question that I did before. She said, yes, we know, but how do we know? Then she finds a forensic toxicologist, and they started working together with an archaeologist, who is in the middle, and he is the male of the group, and that is why he is small, as you see. And the forensic toxicologist is a scientist that works with tiny little amounts of material. Therefore, the anthropologist imagined that this scientist can work together with the anthropologist, and the two together will be able to find out whether there is some material in the toxons or there is no material in toxons. This was a challenging question, and they did find the answer. And today we know that people really were using materials that were going to kill animals by toxic effect. Here are some of the examples that are not that old. They are coming from 4,000 Egyptian excavations. These are the arrows that have been studied by the toxicologist and the archaeologist and the archaeologist. Now look at, even we have the pottery that has been used for putting in the toxic material and use it for killing animals. Here are some other examples from where, as you can understand, it's not very easy to work with. And that is why the very sophisticated instrumentation that we do not have now at our disposal, people are able to answer questions that are extremely interesting and important to the field. Okay, here is the plant. You know, it is quite interesting because people at that time were in a position to find out which plants are eatable and which are not, which are toxic and which are not. Don't ask me how they were in a position to know that. Most probably trial and error, but finally they knew how to prepare toxic materials to be able to use. And by the way, the very big animals were not being killed by the amount of toxicity that the toxins were giving, the arrows were giving, but anyway, the animals were losing their strength, and they were finally, it was easier to be hunted. Therefore, this was very important. Now, I wanted to organize my presentation in 3 different periods of time. The first is the toxicology, which is today being analyzed by the technology that we have at our disposal. And I wanted to show you the collaboration of the different fields of expertise, which is the main topic of this conference, as I can tell from all the presentations that I have seen. Now, coming to 400s, 300s, 500s BC, and I'm talking about Socrates. The very, very important story that has even many scientists and many artists were impressed and created artwork related to the death of Socrates. We all remember that he drank, and you can see that in that picture, in that painting, he was poisoned. He was self-poisoned. And I will be able to change this slide. Yes, with conium. This is the very nice flower that is very effective as a toxic element and kills people. Now, there is a very interesting discussion about whether, yes, he did, or he was poisoned by conium, or he was not. This was also very interesting for me because people are discussing and saying that no, probably he did not, because conium is a neurotoxic material, and the symptoms that are seen because of this toxicity are not the same with those that have been explained by Plato regarding the final minutes of Socrates. Now, this is a question mark, and it's going to be a question mark definitely. It is said that it is written this way to make this death more noble. And it was not with conium, because conium is not like that. It kills differently. Well, I don't know. I cannot say anything because we have no evidence. What we have is written a long time later, the death of Socrates, and we do not know where to rely on this discussion. Therefore, this discussion is not present for me. Anyway, what we know and what is said is that this was the material that Socrates was given, and he was poisoned. And finally, I come to my man. He is Mithridates, and this is a very important and powerful king in northern Turkey and Thrace. He's very strong. He's against Romans. There is a possibility that 50% of his blood is Persian in origin, and he fights with everybody to be able to create this kind of an empire. So every purple thing that you see around is his area at his time. Later on, everything is abolished. But the point is that I don't care with everything that I told you. The very interesting thing is that Mithridates' father was killed by poison. They killed him, they killed his father, and they poisoned him with arsenic. Mithridates never accepted that, or he was always very angry related to that death of his father. And he is the person that started the methodology that even today is called Mithridatism, to try to teach the body, teach the human body how to stand arsenic, specifically arsenic in this case. But this is a methodology that later is used for many things. We give a tiny little amount of some material to an organism, and the organism reacts. But if the material is very small in amount, this reaction is tolerable. Otherwise, if the amount of arsenic that is given can kill a person, and it's a very vital biochemical process that is stopped by arsenic. But if you get used to it if you are becoming used to live and react with small amounts, then you survive. Now, this is the first time where this kind of a medical, let's say, experimentation occurred at that time of Mithridates. Now, so immunity to poisons was developed or writtenly developed by Mithridates, and you can see a publication that deals and writes about this way of immunity gaining achieved by giving the poison itself to somebody to be able to overcome the results and survive with it. So what I wanted to say today was that we have to have the collaboration and cooperation of different fields to be able to get even things that we know that existed 30,000 years before present, to show you or to remind you that people are discussing things that cannot be discussed related to poisoning, toxicology, and human death. And finally, to show you that even an emperor who is very strong and his mostly important thinking is politics, can develop things or put the basis of different things that can be used, are being used even today. So the presentation was based on these three things, and now I'm expecting your questions. Any questions? Anything you want to learn? Anything you want to ask? No questions? Let me ask a question. Thank you.
Thank you.
President.
And now, Gabriel Petridis, in collaboration with Seidas and Demoscan, they present the concept of fluorescent sciences next involved. A question if it exists.
Thank you very much, Professor. I know you're tired, and I can promise you one thing: I have nothing technical, not much. Only that it's going to take me 1 hour and a half to explain you the whole thing. And now that I got your attention, I can go on. I was just joking. It's not going to take so long. I can go on with that. Thank you very much. So the question is, what happened in the ancient world about forensic sciences? I'm a forensic scientist, and I have my colleagues here today, my professor as well. And my interest here is not technical, but I tried to dig up about the idea of forensic sciences, of the evidence, of the proof as we know it today. So how far back in history can we go? All right, sorry, didn't follow. All right, so it's kind of, I'm looking for an archetype, an idea or a person. And so I think I got here story, myth, history, all intertwined. All right. And especially about forensic sciences. So here's my problem. I'm not a historian. So in fact, I have many questions of my own to ask to archaeologists and historians. But then again, from whatever we have, this is the person I want to start, and I really want to tell you that I'm kind of ashamed that didn't occur to me that this person might be the forefather of the forensic sciences as we know them. Because he did something, and most probably most of you, if not all of you, already know. But what I found before that person is so much about taking oaths and some kind of dogmas and beliefs and some traps. And I can give you immediately a nice example of a certain civilization that will try and find out who's the thief by employing a certain kind of experiment. They would put people in a barn, dim-lit barn, and there are donkeys inside that barn, and they would put some soot to the tail of the donkeys, and they would tell these people, you gotta grab the tail of the donkeys, and the donkeys can sense the thief, the criminal, and if they can, and the moment they can sense the criminal, they would bray the donkeys. So when the people come out, they would look at their hands, and they will see some of the people have dirty hands because of the soot as they touch the tail, and some they didn't touch the tail because their hands are clean. So they would decide that the people with the clean hands are the thieves because they didn't touch the tail of the donkeys because they were afraid that the donkey will bray and the thievery will be discovered. So how much scientific is that? So no, this is not what I was looking for. I was looking for, as we define forensic sciences today, scientific proof based on contemporary technology and science. So this person here, maybe some of you can recognize him, is none other than, Archimedes. Okay, this is our guy. Right now, I don't know what else the archaeology and history can find out, but he seems to be one of the most important forefathers of the forensic sciences, because I like that version. There are so many other versions about buoyancy and ship making and everything. So I'm aware there is a discussion between historians about what he did, but that version is what fits me right now to find an archetype. And this is the famous story about the crown of the king. Now, the king had his reasons to be suspicious that the smith who made the golden crown stole a bit of the gold for himself. So he tasked Archimedes to find out whether the crown is pure or not. And that was troublesome for him until, as you know the story, he all of a sudden found out about the water displacement. So much water will be displaced equal to the volume of the matter we submerge into the water. So what's the difference? The difference will come from the weight of the same volume, but the weight will be different if the matter is different. So most probably, he tried same volume but different mixtures of silver and gold. And bad news for the king, even worse for the smith. The history says he realized that was a fraud, a forgery, a thievery. The golden crown was not pure. So he also established something very important for every science. Today we have that of the density, how much of mass we have for a certain value, and so we can distinguish between different matters. What kind of what is the object is made of, and also another very important person you already know, and of course he was mostly famous with his oath, and most probably because he's different, we think from his contemporaries and his ancestors, because he was as much scientific as possible for his own era. Now, of course, I know there is also another debate. How do we know the work of Hippocrates? All right. People are talking about it, specialists are talking about it. And was it his students and everything else that brought us his work? But when you take a look at his work, you can find so many things in medicine that are still the same today, and so many, in fact, ideas. And it's interesting that most probably, maybe we needed only to develop technology to be able to employ these ideas today, but they are kind of quite ancient. So, and once again, as a forensic pathologist, he's talking about trauma and poisoning. And to those who are not very familiar with the concept of forensics, and especially forensic pathology, it's mind-blowing, because he's talking about the origin of death, which means, I can go with the examples to make it a bit more easier to explain it. We have a person with severe head trauma. Now the question is, did he fall? Did he trip? Or did somebody hit him on the head with a rod? Or anything else? So was it intentional? Was it a homicide? Was it an accident? Or did he just jump to commit suicide? That's the origin of death. So a forensic pathologist needs to go first and visit the mechanism of death, which is the biological explanation of death. Now, right now, it doesn't matter. Was it the rod? Did he fall or did he trip? So kind of a same trauma. So the forensic pathologist would start there, performing an autopsy. But he wants to reach the origin of death. Is somebody responsible for the death, and who is that person? So we see at Hippocrates that approach, that he's trying he's starting from the mechanism of death in order to reach origin of death. And that person is not ancient. He's not from he's 2 millennia and some centuries after these people. Do you know that guy? Do you know that person? He's O.J. Simpson. And I'll try and use his trial, which was very intense and famous at the 90s. If you are familiar a bit with the case, O.J. Simpson was a very famous football player, American football player. And it's kind of difficult for me to give that in a nutshell. What happened in that trial, I'll try my best about it. He was prosecuted that he killed his ex-wife and another male person. They were found in the same house. First scene of crime, second scene of crime, the house of his own house, and another scene of crime, his car. So there were so many evidence, especially blood, but not only fiber and so many other objects incriminating O.J. Simpson. But he walked away from the court. He was declared innocent. Not exactly maybe innocent, but here's what happened. His lawyers, the defense, scattered apart every bit of evidence because it was not collected properly from every scene of crime. They took everything away. It was not collected properly, which led them to say that he was framed because he was a black person. And all of a sudden, they find out and they showed to the court. I can't say they proved, but they showed to the court that one of the investigators, one of the police officers, he was racist. So everything fell apart. And there is also an ongoing, no, it's not ongoing right now, but people are still talking about it. It was very recent, another very famous trial of very famous two people, and so much disinformation and misinformation. Why am I talking about these trials? Because here's the question: do you think that what happens in the courtroom is necessarily justice? That's the question. So we have the word trial, justice system, and I'm gonna take you to the word apology and then back to ancient times. So here's the thing: O.J. Simpson or any other trial, the courtroom is very technical. All right, two powers are clashing in the courtroom, and who wins? Does the justice win, or is it another answer here? So, Professor Kalfoğlu talked about his poisoning, but I'm gonna talk about very, very shortly about his trial, about his apology. Now, this is a very interesting word, apology, as they use it in English. So when a native speaker of English says to me, I apologize, I just want to say, okay, go on, I'm listening. Because apology doesn't mean that you are sorry. All right, apology is to account for what you did and explain what you did and why you did it. So, you know, they tried Socrates, and okay, we can talk about later about the outcome of the trial. But in his apology, a very important concept, I believe, was it was there. I can only translate it from Greek to English as judicial competition or judicial challenge. That's how he talks about the trial. And the word try, if it derives from trial, from the word try, is not so far. Whose challenge? Prosecution and defense. Who is going to win that fight? Whoever is the best. All right. So in a few sentences left for my time, that sword is hovering over our heads to do our jobs right. Forensic people, prosecution, defense, judges, juries, in order to be able to say that we can reach justice in the courtroom as long as we can, and we can do our jobs to the maximum, using the contemporary scientific methods and methodology and science. So that alone in the apology of Socrates makes me think that the idea of the fight between the prosecution and the defense is a very old one, and unfortunately, after at least 2 millennia, we are still struggling with that. So thank you very much for your time.
Thank you, Professor Petridis. Any question, please, after this nice piece?
Thank you very much.
Okay. Thank you for your attention. We finished this session about the history of medicine, and
it is the end of the day, but not yet. We are going to be together for the dinner that we have tonight. We're going to meet at 6:45, right in the entrance. So we'll be together here. You can have your coffees or you can be around. The bus is going to come and pick us up at 7 just. Therefore, let's be outside at the entrance, because the bus can come only to the entrance and not within the museum. So let's meet at 6:45, or let's say 6:30, at the outside point at the registration, let's say, desk. Now this is for today. Tomorrow morning, we are meeting at 9 o'clock here for the scientific trip that we have. And tomorrow afternoon, we are going to come, well, tomorrow noon, we're going to come here, have our lunch, and then come here to this room to see the lecture that we're going to, the very interesting lecture about the excavations that we had in Istanbul. The archaeologists that participated to these excavations are going to be with us. They're going to explain their job, what they did, what they found out, and you are going to ask them questions and get the correct answers. So let's meet at the entrance in a few minutes. Thank you. See you tomorrow.